welcome you all to this is APP 13th Annual International Convention as well as the 7th Indo-Australian Conference. As our convener has mentioned, the theme of this conference is emerging trends and opportunity in drug discovery and development. I would like to thank the host institute, Vijay Institute of Pharmaceutical Sciences, for this wonderful. I have visited that place many years ago, and I know um, their contribution to this particular um, area of pharmacy is outstanding. And again, thank you all for hosting this wonderful scientific forums, which is in a hybrid model. And I also will take this opportunity to introduce one of the distinguished scientists and scholar, Professor K. Parang from Chapman University, Irvine, California, USA, who has graciously agreed to join us as the Vice President of APP, American International Branch. Give him a please warm welcome, Dr. Parang. He's a very distinguished scientist. So we welcome Dr. Parang to our esteemed organization, and thank you for your for joining us. Um, as the convener has mentioned, I don't want to spend too much time. This drug discovery and development process is a very complex process, starting from the identification till the drug development, all this which includes all these different pages, clinical and as well as all those page 1, page 2, page 3. And also after the drug will be um, approved by the any of those regulatory affairs, phase 4, which will be again another process, which is again a long process, very complex, time consuming, expensive. And also it has been mentioned that lots of other AI, machine learning, and other processes has improved this complex process, and and make it this process a little bit shorter, less time consuming. But still, then you as a pharmaceutical scientist or as a student who want to be a pharmaceutical scientist of the future, still your presence is very important in this process. No matter how AI will do, whatever the machine learning will improve, but human touch, but your scientific knowledge, your scientific presence is very essential for this process. And as we know, the challenges of this drug delivery is not related to only small molecules. We are talking about monoclonal antibodies, we are talking about proteins and peptides, and many more complex doses from a drug delivery system. So um, I'll strongly encourage you, please be part of this noble profession, work hard to improve this profession and produce some new drugs in the market that will be helping the humanity of the future. And again, as I have told you in the past, one of the things this pharmaceutical scientist has given is this monoclonal mRNA vaccines for COVID-19 that this whole humanity will never forget. So your contribution to this field will be in the future will be immense. And I take this opportunity to thank you all and giving me this opportunity, Dr. Dahia and others, for giving me this opportunity to talk a few minutes about or inaugurating this very important convention. Thank you all.
Thank you, Professor Das, for your comments. And you are well leading the American International Branch, and nowadays a lot of persons want to join that. So thank you for your leadership. And I hope APP American International Branch will grow under your leadership a lot in the upcoming years. Thank you for your comments.
Thank
Now
you.
over to the convener and the co-conveners, they can introduce the speaker one by one, and so that the scientific session can be carried out.
Sure, sir.
A very good morning to everyone. Myself, K. Sunita, Assistant Professor, Department of Pharmaceutical Chemistry, VIPW. Let's move on to the scientific session. I would like to invite Dr. B. Purnima, HOD, Department of Pharmaceutical Analysis, to introduce the profile of our speaker, Dr. Sunita Dahiya.
A very good morning to one and all. Myself, Dr. B. Purnima, working as Professor and HOD, Department of Pharmaceutical Analysis. It gives me an immense pleasure to introduce Dr. Sunita Dahiya. Dr. Sunita Dahiya is an Associate Professor of Pharmaceutics and Drug Delivery at the University of Puerto Rico School of Pharmacy, Medical Sciences, Campus, USA. She holds a PhD from Gautam Bodh Technical University, Uttar Pradesh, an M.Pharm in Pharmaceutics from Rajiv Gandhi University of Health Sciences, Karnataka, and a B.Pharm from AR College of Pharmacy, Gujarat. With over 2 decades of experience in academia and research, Dr. Dahiya directs a lab dedicated to designing innovative, immediate, and controlled drug delivery systems to enhance therapeutic efficacy. Her research has been supported by grants from the Puerto Rico Science Trust and the National Science Foundation. She has published over 50 reviewed articles and 15 book chapters with renowned publishers. Additionally, she serves as the editor-in-chief of a scientific news magazine by the Association of Pharmacy Professionals and actively reviews manuscripts for the leading international journals. Since 2011, Dr. Dahiya has been the General Secretary of the Association of Pharmacy Professionals and is a life and fellow member of numerous professional organizations including APP, IPA, APTI, IPGA, IHPA, SOP, ICCE, APPS, and AACP. I welcome you, Dr. Dahiya, to take over the session. Thank you.
Thank you very much, Dr. B. Purnima, for the introduction. I would like to take this opportunity to thank Dr. Padmalata Madam for hosting the APP 13th Annual International Convention. And I'm also very thankful to the Professor Alekha Das, who is leading the American International Branch. And I would like to thank Professor Parang for joining the American International Branch, and a very warm welcome I would like to extend for his joining. So really we welcome you heartily and hope the branch will grow well in your leadership. And now I would like to share my presentation. And so are you able to see this?
Yes, madam.
Do you see it as a slideshow mode or?
It is not in slideshow, ma'am.
It's not in slideshow?
Yes, ma'am, it is not in slideshow mode.
Okay. So my talk is about the nanocrystal engineering employing the combined bead milling and lyophilization approaches that we are currently using in our laboratory. So we have a couple of projects which are funded for this research, and I would like to give some of the insights and some challenges or practical aspects that might be useful for the students who would like to pursue this sort of research for their journey during the master's or the PhD program.
Madam, it is not in slide mode.
Not in slideshow mode?
No, madam.
Now it is?
Now it is. Thank you.
Okay. Now, all of us are aware about the challenge of the poor solubility of the active pharmaceutical ingredients, and we always see in the literature that around 40% of the existing drugs and around 70 to 90% of the drugs in the development pipeline, they face the challenges of the poor solubility. And if the solubility is poor, definitely they will have some limitations in their drug delivery. Now, for solubility, there are several other things that we need to discuss, like BCS classification and DCS classification, when we would like to correlate the solubility challenge and how to solve it using different approaches, right? But definitely, insoluble drugs will show the dissolution rate limited absorption and leading to several issues when they are delivered in vivo. So this needs to be resolved by the pharmaceutical approaches. And there are several limitations, like because of insoluble API, if it is unaddressed, then it may lead to low or variable bioavailability, and it may show the high fade or fasted variations, retarded onset of action, lack of dose proportionality, and higher interpatient variation. These are some of the common of the consequences which might appear depending on the type of drug and category of the drug. This type of issues needs to be resolved to have an optimal drug delivery. Now, a very famous biopharmaceutical classification system, which has been adopted around 1995 by FDA, and it was developed by a group of researchers, Amidon et al., and they classified the drugs into 4 classes. So this is very basic that we use to select the drug candidates whose solubility should be manipulated in terms of formulation development or to resolve those challenges. Now, class 1, we know that they are very good candidates, but problem comes with class 2 and 4 candidates. So class 2 candidates are often very good candidates, because if you are able to solve the low solubility problem, they have already high permeability, and you may change by different approaches the class 2 drug into the class 1 drug. Similarly, class 4 drugs may not be showing a good performance if the same approaches that we use for class 2 drugs we use in class 4 also. So they require different approaches to be used. Today I'm going to talk about class 2 candidates and how we resolve the issues with the class 2 using the nanotechnological approaches. Now, there have been several approaches. The most widely used one is the salt formation and pH modification, and the micronization approaches, or amorphous solid dispersions, cyclodextrin complexation, and lastly, the most recent one probably is the nanotechnology. There are some others which might fall in one of this category, maybe the use of functional excipient, etc. Nowadays, several companies are working on that, developing the functional excipient or co-process excipients, which claim to be very good in terms of formulation development, right? Now, coming to the nanocrystal technology, that is the major focus of today's talk, that I would like to say that there are major 2 approaches which are used for nanocrystal preparation or production. Those are top-down approaches and bottom-up approaches. So as the name suggests, when you have a higher particle size and you want to take it to the lower particle size, usually top-down approaches. So you are downsizing the particles from top to the lower side, and that's how the name comes, that is top-down approaches, that mainly the milling approaches or the high-pressure homogenization, etc. Also, the bottom-up approaches like precipitation approaches, usually solvent anti-solvent or solvent removal approaches, or the use of precipitation in supercritical fluid, etc. There are combined approaches or combinative approaches where we need to use more than one technology to solve the solubility and dissolution challenge. For example, you combine 2 technologies like micronization and high pressure homogenization, or you combine spray drying with the HPH, or you combine WBM with lyophilization. So today we are going to talk about WBM, that is wet bed milling with lyophilization. Now, when we come across the nanoscale systems, it's a broad area, right? So it's very important to distinguish between certain terms that we are going to use frequently in this presentation, that is nanosuspension, nanocrystal, and nanoparticle. So sometimes it's very basic, but I would like to say for sort of students who are listening to this lecture, that when you say nanosuspension, usually it contains drug nanocrystals with surfactant or polymer stabilizer around or surrounding the particles. So they are nanocrystals in the liquid medium, and so the nanocrystals are dispersed in a liquid. When you say nanocrystals, usually we mean here is the solid nanocrystals, not nanocrystals in the liquid medium, right? So nanocrystals are the pure solid crystalline drug particles, and the average particle size in this class is 100 nanometer to 1000 nanometer. So you can say that a 0.1 micron to 1 micron. So our target is to bring the particle size below 1 micron, right? Now, dried nanosuspension is another term that can be used usually for the nanocrystals. And when you say nanoparticle, it is very general term. So you can say that all nanocrystals are nanoparticle, but all nanoparticles are not nanocrystal necessarily, because usually when we say nanoparticle, it also can be used for polymeric nanoparticles or the lipid-based nanoparticles or for example, liposomes, etc. Any other type of nanoparticles, which basically use the carriers. Now, how nanocrystals differ from this nanoparticle is that they are carrier-free. So we do not use any carrier here. The only thing that we use to develop the nanocrystal is the stabilizer. Right? So it is a very, very important thing to know that how nanocrystal differs to other nanoparticles. And that's why we should know some of the attributes or distinguish attributes of nanocrystal compared to other nanoscale systems. For example, they are conceptually simple and practically straightforward to prepare and develop, because there is 100% drug, no carrier material. Remember, there may be stabilizer, which is essential to be used and must be optimized using the process development. And if the nanocrystals are in liquid form, that is nanosuspension, it must be dried to fabricate them further as the solid dosage forms. Now, nanocrystals can solve several biopharmaceutical delivery problems, like if the drug is suffering from too low bioavailability after oral administration, or too low penetration into the skin, or if the drug is suffering from the low dermal bioavailability, or if the drug required a much larger injection volume and the target is to formulate the intravenous injection. So in all these cases, nanocrystal is really a helpful approach for the development of drug in this type of delivery systems. Now you can see here in this figure the sum of the carrier that we already discussed, which are distinct to the nanocrystals. Like it doesn't use any carrier, it has size below 1 micron. So a particle size range is really big. It's not something necessarily that will use a particle size below 100 nanometer. Right? It show enormous surface area. It show much enhanced saturation solubility and increased dissolution velocity. It may have crystalline or amorphous structure based on what method has been used for development of the nanocrystal. Also, it shows the increased adhesiveness to the surfaces of the cell membranes. So these are some of the attributes that we should consider while we take this approach. Now, the biggest advantage or attribute is the enormous surface area, right? So very fundamental of the pharmaceutics that we all know probably, that when you reduce the size of the particle, the surface area increases. Now, how much it increases depends. And that's how the role of wet bead milling comes here, right? So you may have other approaches like micronization or use of the dry milling, etc., but they may not be as effective as this technique when your target is to produce the nanocrystals. right? So there are other milling approaches that can be used and have been used since several years in the pharmaceutical industry. But this is where you want to reduce the particle size to the nanoscale. So as the particle size is decreased, the surface area will be increased, and that's how there will be a much more enhancement in the solubility and dissolution. Now you can relate this with three of the major advantages, like reduction in the particle size. You can see here in this slide, taking example of a particle with average size of 100 micron, and when you are able to reduce it to 0.5 micron, that is roughly 500 nanometers. So this will increase the specific surface area, right? And how this will increase the particle curvature to give more of the dissolution pressure, right? And also it will decrease the diffusional layer path length, which is one of the very important parameters, because the H that we use for the Noise-Whitney equation, if you recall your, if for the sake of students, if you recall your first or second year physical pharmacy principles, you might be knowing, and even in biopharmaceutics, that how we calculate or how do we relate the physicochemical property and dissolution to the rate of absorption, right? So H is considered to be the distance between surface of the drug particle to the bulk of the solution, and it typically represents the distance over which the drug must diffuse to enter the bloodstream. So it's a kind of representative of the distance required to reach from the surface to the blood, right? So it definitely affects the absorption or the entry of the drug particle into the blood, right? So this is how the nanoparticles will increase the dissolution velocity, which is very, very important parameter to relate with the absorption, which occurs by passive diffusion. Now, the milled particle, right, because of the small size and enough surface area, they will have high surface free energy, and they will also show very much improved wetting and property and solubility, which is one of, again, the challenge for hydrophobic drugs, right? And many of the drugs fall in this class. So it's really important to improve these physicochemical properties to get enhanced dissolution. So there is not only one thing that is reduction in particle size, but there are also associated attributes which lead combinedly to the enhancement of the dissolution. Now also, nanoparticles will give the better oral bioavailability, because it will give the increased adhesiveness to the because of their smaller surface area. So usually for the oral delivery, that displays increased van der Waals forces of attraction between the nanocrystal and the gut wall, that is the mucous membrane, right? And it contributes to the increased drug adhesion and bioavailability, because they can quickly penetrate into mucous gel layer, right? And that's how they can improve the bioavailability, right? So this is how not only dissolution, they also contribute to increasing the absorption, not only dissolution rate limited, but also by improving the drug retention into the mucous gel layer, right? So this is the mechanism how they enhance the bioavailability. Now coming to the technique of the wet bead milling, right? We do not use, another biggest advantage is almost a green method, environment friendly method, because you need not to use any of the harsh organic solvent, or you need not to use extremes of the pH to solubilize the drug. You can use high API concentration. That means the drug loading can be really high in this curve, where other nanoparticles struggle to have high encapsulation efficiency, right? This approach is scalable. So if you use some of the methods like QBD and you optimize the process, it really can be scaled up for the commercialization. The method is really reproducible with optimizing the parameters, but the parameters required to be optimized on case-by-case basis. So for one drug, if I optimize the parameter, not necessarily it will work for another drug, right? Now, there are wide variety of the hard, dense milling media, right? So milling media, in simple terms, they are the bowls. You might be heard about the bowl mill or dry mills, right? So they are media or balls which are used for size reduction. So the material which is made up, which is used to construct this media is very important because they are in intimate contact with your drug material. And when you develop a new formulation, the contamination is really important, right? So usually the yttrium stabilized zirconia oxide beads are very, very widely used and safe to use and reusable for the pharmaceutical material because they find almost no interaction or contamination with the drug that you are using in your experiments. Now, it's very important to choose the correct particle size, right? So sometimes we think that we should take very big size of the balls, or we should take a medium size of the ball, or we should take a correct size of the ball. Now, again, these are the process parameters, what we call as the critical process parameter that must be optimized by a systematic approach, right? But in general, if you use two small bits, right? For example, bits like 0.03 millimeter, right? They are very much lightweight and they cannot generate sufficient energy for the particle breakage when they impact the drug particle, because here the mechanism is impact and attrition, right? So they are so lightweight that they may not influence the impact that is required, right? So they may not be suitable for milling. And sometimes we use very big particles, so they have increased collision frequency between drug particle and bits, right? And they will be able to produce the finer nanocrystals to be produced. Now, ideally, the bead size less than 0.1 millimeter to 20 millimeter are typically used, right? So you can use 0.1 millimeter beads or 0.3, 0.5, 0.8 are usually available, right? Now coming to the mechanism of the wet bead milling, right? We know that the mill will be operated at certain speed, right? Now the speed that we choose is very, very important because it's not always that you use the maximum speed that is available with your equipment will give you best results. It's never like that, right? So you have to really work on what should be the milling speed and milling time, right? Now, if you use a very low speed, it will create the low angular velocity, and you can see here the movement of the the balls, right? So they will move along the jar, right? So that will not give a very good particle size reduction. At the same time, if you use a very high angular velocity at a very high speed, the balls are thrown out to the mill bowl because they will have a very high centrifugal force and there will not be any size reduction because the impact function will not be effective in this case. Now coming to the ideal speed for the mill, it should be usually around 2/3 of the critical angular velocity, where centrifugal force occurs at its maximum, giving the cascading movement. So this is the speed which usually give you the best particle size reduction. And we have experienced this principle in our laboratory when we were using our equipment from the RECH. The 100 model. And when we started the experiments with the highest speed, we were not getting good results. And then we came across this principle that always using highest speed will not work. And you really need to optimize the speed and the time. Now coming to the another very, very important aspect is the stabilization. Without using stabilizers, the nanocrystals cannot be developed, right? Again, these are very fundamental of the colloidal chemistry, where you might be knowing the mechanism that are imparted by different type of the stabilizers, right? For example, electrostatic stabilization based on electric charge developed on the surface of the small particles or nanoparticles. And also the steric stabilization, which depends on the physical presence of the polymeric material or the non-ionic material or stabilizer that you are using, right? So although the purpose is the same, but the mechanism, how they give how they develop the repulsive forces over the surfaces are different. You can see here several examples of this. And this needs to be optimized, because if you think that, okay, this drug I saw in the literature, and for this drug, sodium dodecyl sulfate was very effective, and it will be effective in my case also, for my drug also. But no, it will not. It may give you the worst stabilization probably, right? So you need to look into the chemistry of your drug and chemistry or mechanism of your stabilizer, and what is the size range, what should be the stability and redispersion that you are targeting to achieve, probably what solidification method that you are going to use after the milling method. All this matters when you choose the stabilization, because stabilization also affects other critical quality attributes like dissolution or solubility, not only the size and charge, right? So you may have steric stabilization with the polymeric stabilizer or with the non-ionic surfactants. Now, there are advanced wet bead milling technique that probably was developed by a company in Japan, that they use the ultra apex mill like Kotobuki Industries. They tried the centrifugal technology, which is inbuilt or integrated into the milling equipment, and they use the milling media of very small size, like 15 to 100 micron, not millimeter, right? And they used this approach for several drugs like albendazole, danazole, omeprazole, probucol, etc., and to enhance their dissolution and absorption properties. But this cannot be used, because in this cannot be used for a normal mill. This is a specialized milling equipment in which the separation of the milling media from the material is inbuilt, right? Because you may not have sieves to separate the milling media from the material, right? So this is an advanced version of the wet bead milling. Now, this is the equipment that we are using in our lab, and this is my graduate student, Cassandra Ruiz. She is working with the project. This is her PhD project. All right, so this equipment is a very compact and benchtop equipment. We use single jar, although there are provision to stack more than one jar. It's available for use of one jar, 2 jar, or 4 jars even. It's very versatile and flexible. It can be operated at varying speeds, right? But definitely for smaller batch size only, you can use that. Now, the mechanism here is a little bit different because it's a plenary ball mill, and it works on the principle of friction and impact, where it generates very high centrifugal forces and very rapid particle size reduction. So you might be thinking that why can't we use ball milling? Remember, when you target the nanoscale particle size, those approach like dry milling or using another mills or mills which are using the bigger media will not give you this particle size. And that is how this mill is unique in achieving the nanoscale particle sizes. Here, in this case, you can see here the sun wheel, right? And this jar, which is located on this circular platform, which is known as sun wheel. And when this sun wheel rotates once, this jar rotates 2 times in the opposite direction. So this movement will be clockwise to anti-clockwise, and that's how it says that it works in the -1 to 1 to -2 ratio, right? So this minus denotes the clockwise and anti-clockwise movement of the sun wheel with respect to the jars. Right? So because of this movement, there will be really a very impactful particle size reduction because of the movement of the balls when the jar is placed on this sun wheel and when they are rotated with this mechanism, right? Now, these are the mill accessories. You can see here 1 millimeter balls, 0.8, 0.5, 0.3, and even you can have 0.1, right? So these are very high density yttrium stabilized zirconia oxide media, right? And you can load this media. You have to definitely find out that what should be the volume of the media, right? For example, if you have a 50 milliliter jar, what should be the volume? And you have to convert the weight of the media to the using its density to the volume, right? Because it will be in form of a dispersion. When you load your material with stabilizer, with drug and with milling media inside the jar, and then you have to put the jar, you have to weigh the jar to make a balance within the mill, and then you have to process the mill. Please note that I'm not putting here all the pictures because this is a funded project and we have applied for the disclosure. So I am giving here very less data that we have generated, and some we have been advised not to show all the things until it is under the review, right? So that's why I am trying to explain something, not by giving pictures or real pictures from our laboratory or the data that we have generated in our laboratory. So I'm sorry about that. But if you have any question further, you can definitely contact me, right? So you can see here the size, right? So as the size goes down, it's really hard to handle them. And these are very, very costly, definitely. A 1 kilogram costs around $1,000. And the cleaning process is also very special and use a lot of solvent or for reusing this material, right? Now, this is the process or mechanism how you use this media and how the stabilizer will be coated, will be making a coating or a thin layer surrounding the smaller particles, and they will give them the stabilization or a kind of repulsive forces that we usually measure with the zeta potential when we measure the sample of the nanosuspension with the zeta potential. Then we will be able to know that how much stabilization we are able to achieve, right? Now, these are some of the very important process parameters which affect the quality of your product, like milling time and milling speed. So you have to make a balance between them, milling media, drug concentration, right? So if you are using very high drug concentration, maybe you will not be able to achieve a desired particle size, right? Also, you need to optimize the milling media, what size media you should use, right? What size is and also the concentration of the surfactant. So two things are very, very important when we talk about stabilizer. One is the type of stabilizer and another one is the concentration of the stabilizer. Sometimes we have experienced that one stabilizer may not be effective to provide an adequate stabilization. In that case, you need to use more than one stabilizer and you need to optimize concentration of each one, right? Here also, in this table, you will see that the experimental batches. Remember, again, I'm not disclosing all the data here. I have put here some of the batches that we prepared and we did not pass the quality criteria when we use it in the zetasizer in our lab. So you can see here various ingredients, that is stabilizer to the drug. I'm also not disclosing the name of the drug for that reason, right? So you can just consider it's a drug. You can say HPC, the sodium dodecyl sulfate, or TPGS, etc., various stabilizer that we have used with respect the drug concentration. Right? So you can see the particle size we achieved, right? But the quality criteria were not passed. So we were not supposed to use this type of combinations to proceed further with the formulation, right? So we did a lot of experiments, knowing what will not work rather than what will work, right? So this is how step by step we reach to our optimal batch after at least 35 trials. Right? So you will find that only out of these 13 batches that I have shown here, out of all these 13, the satisfactory in terms of passing all the quality criteria with the particle size measurement, only one batch passed, and another batches like P1 to P11 did not even pass the quality control criteria. Right? So we need to move on another parameters, varying the parameters, like you can see here, not only the ingredients, also the process parameters of the mill, like mill speed or time, media, what was the bead, what were the loading of the bead, that is volume of the bead which was used. these are some of the batches that was prepared by the graduate student in the laboratory. And you can see here the sizes of the nanosuspensions. Right? So these are all of the nanosuspensions that you can see in the bar graph, various sizes. Now, when you have a nanosuspension in liquid form, and then you want to further develop a formulation, a solid dosage, right? Then that nanosuspension will not work. So in that case, you have to achieve the smaller particle size, nanoscale particle size, but yet you want to develop a solid dosage form, then you need to solidify those nanosuspensions. And here is the role of any solidification technique comes. For example, you can use either spray drying or lyophilization, right? So what we use in our lab is the lyophilization. So once you have your nanosuspension prepared, you can now use that. If it passes all the quality attributes in terms of particle size, and the PDI, polydispersity index, and zeta potential, then only you can move to other solidification technique like lyophilization, and then you have solid nano. So these are some of the parameters that you need to know. So a little bit introduction I have given of the lyophilization. Many of you might be using it for different other purposes because lyophilization is a common technique that can be used. But again, here it is about optimizing the parameters when you are using it for the nanosuspension. Because once solidified, it must be redispersible and it is stable. So using the technique is simple, but maintaining or preserving the size attribute is really difficult after the solidification, right? So lyophilization, key principle, you know that you have to go for pre-freezing the product, and then it will be sublimating and removing the residual water during the secondary drying, right? So freezing, primary drying, and secondary drying to have your product in terms of a porous dry matrix, which will ensure the stability, storage, and redispersibility of your nano, right? You may have other parameters. For example, you have to know the purpose of your final product, right? What you need, how much stability, what should be the particle size after redispersion, that is very important. The phase of action, right? And what cryoprotectant and lyoprotectant could be used, right? So usually when you compare, in our laboratory, we compare that what happens if you do not use any lyoprotectant or cryoprotectant in the laboratory, then what happens? Are your nanosuspension able to preserve the size that they already have after milling? So the answer was yes and no, because it was not able to preserve the size attributes, right? So redispersibility was not achieved unless you use the cryo and lyoprotectant, right? So usually cryoprotectant preserve your material from freezing stage, right? So usually the faster freezing rate is preferable because you freeze your sample really fast, and then you sublime it, right? And then it's very essential to make sure that how much time you keep your product into the lyophilization, how much you keep, because remember, all these are the stress condition, and they will be very much affecting the size and the solid state properties when you analyze them after lyophilization. Right? So performing is another issue, and getting and maintaining those quality attributes after performing a process is different thing, right? So to get the attributes that you target, you must optimize all these parameters. Now, we may have different purposes, right? So before freezing, you can add right? No matter whether it's cryoprotect or lyoprotectant, many of those are used interchangeably. So sometimes we say tree halos, it can be called as cryo as well as lyoprotect, because it will protect your particles at both stages. Right? So usually they are added before freezing, no matter they are lyoprotectant or cryo. They are added before you expose your sample into the lyophilization process. Right? So these are the key critical process parameters like freezing rate, primary drying temperature, secondary drying temperature, or the cryoprotectant concentration, right? Cooling or heating rates, and the residual moisture content, right? So for example, if you think, okay, I see the product is dried after 4 hours, and you just remove. But remember, at that time, you might have the residual moisture content, and it will not give you good redispersibility and stability. So you must ensure that how much time you should keep your sample into the lyophilization to ensure it is completely dried, right? So these are some of the parameters and challenges, right, that must be optimized. Now coming to the lyophilization procedure, you can see here some of the pictures like pre-freezing, the nanosuspension at -80 degrees. So depending on the sample, right? And also during the freezing, it's very important that both shape and size of the vial that you are choosing for lyophilization or for freezing, right? Because it starts from upward to the downward. So if you have a very higher sample size in the vial, maybe you have drying on the upper surfaces and your lower side material will not be dried properly. So that is another challenge. So there's also a very small thing, but so important in terms of the stability and redispersibility of your final product, right? So all these are some of the insights into the process that you must account for when you work with this type of procedures, right? So you have to then place the frozen sample into the lyophilizer. Right? For our case, we use around 18 hours to make the sample to complete dryness. Right? So then you have to again take and make a sample out of your lyophilized powder and further analyze. Now this analysis will tell you whether we need to change the particle concentration in this case if you are not able to achieve the desired particle size after redispersion. Right? And then you have to keep on tracking what is the particle size after 1 month, 2 months, 3 months, for example. Right? So to ensure its stability over the time. So this is the procedure that how you redisperse it and then analyze it again for the particle size parameters. You can see here the particle sizes. We were considering only one batch, right, which gave us 286 nanometer particle size, right, within 120 minutes of the effective milling. That is 2 hours of the milling with 5 minutes of the post time after each 30-minute cycle. That was the milling parameters that we set into the lab. So this is the batch 13 that we used TPGS for this particular batch. Also, to see that whether the milling is inducing any changes in terms of its crystallinity and amorphicity, we carried out the crystal powder diffraction study, and you can see here the diffractogram of reference drug, the sample drug, right? So this reference drug diffractogram from the literature, sample drug, the sample that we purchased, and the 2 batches that were passing the quality control criteria, right? So you can see here that mostly, I will not say 100%, but mostly the crystallinity was maintained, right? So our target is nanocrystal to have smaller size but crystalline particle. Our target here is not to change the particle from crystalline to amorphous, right? So we were able to preserve the crystallinity. So there was basically no mill-induced amorphization in this case, the parameter that we use. Now coming apart from the laboratory work to the general aspects of the nanocrystal, which are in the research or in the market, right? So this technology have a very good market potential because they can be amenable to different routes of administration, right? And several applications have been submitted, several are under clinical trials or they are under development, right? So it can be used for several routes, oral, intravenous, intramuscular, ocular, or intraperitoneal, and even intratumoral, right? Although less work has been reported for the surface functionalization or the targeted drug delivery using the nanocrystals, right? You can also see here one or two case studies that I have taken from the references that you can see below on the slides. One case was of the aprepitant, which is a cancer chemotherapy to relieve the cancer chemotherapy induced nausea and vomiting. This drug, you can see here that how much the plasma concentration was improved using this technique when the particle sizes were reduced to 120 nanometer, right? So as the particle size was reduced from 5.5 micron to 1.8 to 0.8, so you can see here the clear difference, not only in the in vitro solubility and dissolution, but also in terms of the plasma concentration. Coming to the bioavailability, right? So usually it's very good in terms of having no effect, or no fade or faster state effect, right? So one drug which one company was working with developing this effect with the drug magistrol acetate, right, which is an appetite stimulant drug. And with clinical trials, they found that compared to the original suspension, right, the dose was reduced significantly. So that was a very a good achievement in with nanocrystal, because you were able to reduce the drug dose, right, which always desirable. Some of the other marketed or FDA approved dosage forms by wet media milling, I'm showing here. You can see all the brand names and the API, that is active pharmaceutical ingredient, what dosage forms, indication for which the drug have been used, and what is the innovator company launching these products. So the concluding takeaways is that the nanocrystals can be used for both the existing drugs or for the new chemical entities to allow for the lead selection strategies based purely on the efficacy and safety. For example, if you have a new chemical moiety out of your drug discovery experiments and you think that the drug is very potential, have pharmacological promise, but in terms of its physicochemical properties, it's challenging. So in that case, this technology can be used. Also, nanocrystals are a proven approach which has been embraced by both academia and industry, and there is a lot of research that you will find on this area. Also, the combined approach is always welcome, because in that case you will not just target the liquid, but also you can move your nanocrystals to the solid dosage, right? However, mitigating challenges with each of the process that you use is very, very important. So you must optimize the methods on case by case basis. It cannot be generalized for each and every drug, although the approach is uniform, right? Now, it has very good future potential. As I told you, that nanocrystal has not been much used for the targeted drug delivery. Rather, it is mostly used to overcome the limitations of the solubility and dissolution, and that's how you can improve the oral bioavailability. This is our project team. One student who is working on this project is Cassandra, and these are the co-PIs, Dr. Hernandez and Dr. Torsten Selzer. He is responsible for solid state analysis, and Dr. Hernandez looked for the cellular permeability studies of the nanocrystals. These are some of the funding that I would like to acknowledge the funding agencies for providing us the fund for this research. Right? We have other funding mechanisms also that we are using as a resource for this project from the National Institute of Health, or from the National Science Foundation, from the local bodies like Science Trust in this territory, and internal grant, of course. So all these are the funding which we have used in developing this project. If you have any questions, you are welcome.
Over the
Hello.
judges. Thank you, Dr. Daya.
Hello, good evening, ma'am.
Yes, good evening.
Yeah, I'm Dr. Kiranmai Mandava, Principal of St. Paul's College of Pharmacy. Happy to listening to you, ma'am. So many I have listened to, and this is one more thought-provoking session. So one small question I have, ma'am. It's very interesting actually. You have clearly mentioned that this particular technique, what you have emphasized is it's not the novel thing, but to improvise the solubility and dissolution of the existing formulations. Am I right, ma'am?
Yes.
Yes, ma'am. So, how to maintain that crystallinity, ma'am? What is the specific uh, clue you can give as a renowned researcher to maintain that crystallinity without disturbing its morphology?
Yes, because usually, remember, as I told you that I have not covered several aspects here. To maintain the crystallinity in the nanosuspension is really easy, right? It's easy when you optimize the parameters, but again, you have to take several trials, right? You have to take several preliminary trials, and then you use any systematic approach like design of experiments, etc., to use those trials and see that what are the best combinations. Right? In that case, I would say that maintaining the crystallinity, you need to see that what should be the size of the media and how much time you should process your material into the mill. That is very important. But because your material must not be overstressed, you have to look at the temperature and pH change after every 30-minute treatment to see that if there is heat generation, probably your drug might be adversely
Yes.
impacted by that, right? So usually, if you optimize all these parameters, you will have crystalline particles after nanomilling. But again, when you solidify it, remember that we were trying to analyze them like DSC and XRD after lyophilization, and we found that techniques like DSC and XRD sometimes will not be able to detect such a small amount of drug. And in that case, in your sample, other ingredients will be overpowering. So you will see the structure of the other ingredients which are in higher amount. For example, if you have added 5% of the cryoprotectant, for example, right? And if you have like 50% of the stabilizer of the 1% of the drug, so you have more amount of other ingredients. Although this technique doesn't use any of the carrier, but you have some excipient, like 1 or 2 excipient, which is really high in terms with respect to the drug. So after solidification, you might not be able to see whether it is crystalline or amorphous, and maybe you will see that your drug have phase transitions.
Yes.
In
Yes,
that case,
ma'am.
you have to go for other techniques like Raman spectroscopy is better to use in that case, or you can go for FT-ATR, or you can go for solid state NMR. Those are
Yes.
more advanced technique in that case to look into the real structure of the drug, because that method is not sensitive enough to detect the structure of the drug where it is below certain percentage in your sample. For DSC, at least 25% of the presence of the drug is required. If it is not there, your drug will not be detected into that.
Yes, ma'am. Yes, ma'am. It's a steric stabilization. It's a really interesting aspect you mentioned about. Thank you, ma'am.
Good morning, ma'am. Myself, G. Krupamayi, Assistant Professor from VIPW. Thank you, ma'am, for such emphasizing session on nanocrystals by using wet bead milling and lyophilization. And you have emphasized on preparation of nanocrystals and also bioavailability, saturation, solubility, such physicochemical parameters, and how we can achieve through preparation into nanosuspensions. Here, my question is, can we use the same sort of techniques like wet bead milling and lyophilization for improvisation and enhancement of other formulations, not nanosuspensions, any other formulations depending on particle size and their stabilization?
No, in that case, remember, this is a specialized mill usually used to have smaller particle size. If you just want to decrease the particle size of your raw material, then you can use any other bricks. Like you can go for micronization. So I don't know really if I answered your question, but if you are talking about the raw material, here we are talking about raw material or raw drug and how to reduce the particle size. Nanocrystal is reduction of the particle size of the drug and then using that drug in the formulation. Right.
Yes, ma'am.
So
Yes.
if you have like something like microemulsion, nanoemulsion, no, it cannot be used like that.
Yes, yes, ma'am. Okay, ma'am. Thank you.
Any other question?
We can continue for the next speaker. Thank you, Dr. Daya, for your lecture. Our next speaker is Dr. Kamal Dua from Australia, and I invite the co-convener to introduce Dr. Dua.
I would like to invite Mr. D. Prasanna, Associate Professor, Department of Pharmaceutical Chemistry, to present the profile of our keynote speaker, Professor Kamal Dua.
A very warm good morning to you all. I'm pleased to introduce the profile of Dr. Kamal Dua. He is an associate professor, Discipline of Pharmacy, Graduate School of Health, University of Technology, Sydney, New South Wales, Australia. And he is the president of APP Australian International Branch. Dr. Dua has research experience of over 17 years working in the field of drug delivery targeting inflammatory diseases. Dr. Dua is also a node leader of drug delivery research in the Center of Inflammation at Centenary Institute, UTS, where the targets identified from the research projects are pursued to develop novel formulations as a first step towards translation into clinics. Dr. Dua researches in two complementary areas, drug delivery and immunology, specifically addressing how these disciplines can advance one another, helping the community to live longer and healthier. This is evidenced by his extensive publication records in reputed journals. Dr. Dua's research interests focus on harnessing the pharmaceutical potential of modulating critical regulators such as interleukins and microRNAs, phytopharmaceuticals, and developing new and effective drug delivery formulations for the management of inflammation in chronic airway diseases and cancer. With these few remarks, I would like to hand over the session to Dr. Kamal Dua, sir. Welcome you, sir.
Thank you, Dr. Prasanna. That's very kind of you. I'm going to share my screen. Just let me know if you can see the screen. Can everyone see the screen?
Yes, sir. Yes, it is
Okay.
visible.
Okay, good. Thank you. You okay for me to start?
Yes, sir.
Okay. Thank you, everyone. And thank you so much for having me as one of the speaker. And thank you to actually Association of Pharmacy Professionals for inviting me to their annual Congress. So the title of my talk you might have seen already, which has been disseminated to everyone. It is around the novel therapeutic interventions in the chronic airway diseases. But before actually I start, I have little modified the title to the novel therapeutic interventions for chronic airway diseases, but we talk about the whole of the discovery and a roadmap, and how particularly in my team, I use the interdisciplinary research, where I not only just work on the developing the nanoparticles and characterizing them, but also how those nanoparticles are being tested to the cellular and molecular level, and both in animal as well as in vitro models. But at the moment, what I'm going to show you is more on an in vitro model and a very little of the animal studies. But there is a very niche thing which you will see in my presentation. Being born and brought up in India, still been very passionate towards the phytoceuticals. So you will see 90% of my studies, they been carrying or containing or encapsulating the phytoceuticals. And I take those phytoceuticals in advanced delivery systems, which we call nanoparticles. I primarily work on liquid crystalline nanoparticles and test it on the different respiratory diseases, particularly asthma, COPD, and lung cancer, and also to the cellular and molecular level. Okay, now I think this is a country where I'm coming from. I born and brought up in India, as I said, but I've been living here for almost last 15 years, which is the state called New South Wales, which is in a green color. Probably you might be seeing with the pointer, and one of the biggest state called Sydney. And everyone know these two hallmark features of Sydney. One is called as a Sydney Harbour Bridge, and the other is called as a Sydney Opera House. And the university is not very far from these two hallmark icons of the Sydney. Our university is just instead where you're seeing these big buildings, instead of going on this side, if you go on the other opposite direction, the university is hardly 15 minutes away from there, 15 to 20 minutes from the Darling Harbour. Now, the school where I'm coming from, this is a university. This is just a couple of buildings of the university. The glass color building is a new addition in our university at the moment. This is called Tower, next to the Tower building, which is a brown color called Building 2. But this is a school where I based at and where our discipline of pharmacy is. Now, in this school, all the glass color buildings are the one which is called as a graduate school of health. And we have all the postgraduate programs starting from Master of Pharmacy, Master of Orthoptics, Genetic Counseling, Speech Pathology, Psychology, Indigenous Health, and Good Manufacturing Practice. Now, this particular school, I'm also been affiliated with some of the research institutes from the University of Newcastle, which is called as a Hunter Medical Research Institute, where I'm on an honorary appointment, and also with the Centenary Institute, which is a part of both the UTS as well as the University of Technology, Sydney, where I'm based at. Now, before actually I start, I always emphasize on this slide in any presentation I do, whether it is a science presentation or a non-science presentation. And this is really important because if you see, it says like learning to collaborate and collaborating to learn. So it is both bi-directional thing. And most of the people, I believe, those who are sitting in this conference on screen and off screen, they would be all college and university professors. So in our current time, it is really important to collaborate because no one is champion in one area. It's always good to collaborate with people that will ultimately reflect on your research track record and also ultimately enhance not only just your reputation, but also the institutional reputation. So this paper by the Baldwin and Chung, it is really had clearly shown that learning to collaborate and collaborate to learning is important for the university professors. It is really important for our own professional development. And at the end of the day, it is also important because when you will be doing a good job in your research, it is ultimately your institution is also getting a reputation as well. And at the end of the day, to do this collaboration, you need to come out of the comfort zone. And particularly, I'm online at the moment with all of you. And this has all emerged out during the time when it was COVID. We all used to attend highly face-to-face conferences. We do attend, the things are getting normalized now in the last 2 years. I have attended significant face-to-face conferences in last year, but it clearly shows that internet had a lot of power. And to do this collaboration, we have to come out from our comfort zone. And to do that, you can use this tool to tag to the people, to connect to the people. I get so many connections on LinkedIn and Twitter, and these are the two main powerful tools which actually can make your professional and growth goes really quite high. So my talk is all around the chronic respiratory diseases. And the question comes, the first important, let me talk about science now. Why chronic respiratory diseases and why we have to focus in this area? And particularly why the emphasis nowadays is going in this area. Now, I'm not going to show too much of the statistics and stuff because whenever I go to any specific country, I usually try to collate the data of that country showing the burden of the respiratory diseases, particularly, and the factors associated with it. So if we really look, most of the chronic respiratory diseases is being an umbrella term. and it comprises of diseases like asthma, COPD, which is also called emphysema or chronic obstructive pulmonary disease, and lung cancer. And there are so many new diseases like pulmonary fibrosis, and there is a new disease which has just been came, which calls ACOS, which is the asthma COPD overlap. The infections are very common, COVID is one of the major infection we have just came out a couple of years ago. There's influenza, there is rhinovirus, there is Haemophilus. They all are respiratory viruses. If we really look at the statistics, actually, there are 4 million premature deaths just happened because of the chronic respiratory diseases. And one of the major factor is the tobacco smoking. And if I really, I not don't want to specifically point out on India or any other country where the usage of the tobacco smoke, and I have that statistics, I have intentionally didn't put here, but it is quite high. And even in both genders, male and female. I presented in Taiwan, and Taiwan also was really very bad in terms of their actually conditions as compared to the other countries. Despite we think Taiwan is a small country, it has really had a good, quite a good number of the healthy conditions, but it is not as the case. And all those respiratory diseases, COPD, not only in Taiwan, it has been prevalent across all the world, including even our Australia, where it ranked third in number. Now, before actually I jump in the reality, as we know, the pollution is one of the major problem. And if the pollution, if we really look at the real scenario, and particularly it come to India when it comes to winters, so you get smoke, which is called as a combination of the smoke and fog. And imagine when a person with a respiratory underlying condition goes out in that condition. So people used to talk about, asthma happened because of allergies, or COPD happened because of smoking, and pulmonary fibrosis is a further, degradation of your alveolar macrophages or the alveoli and stuff. But at the moment, there is a big buzzword called pollution. I presented in Singapore, I presented in Malaysia, and the statistics, this early this year in 2024, I was in University of Groningen, and Netherlands was even worse in terms of pollution, which is one of the European country. So what do you think of the pollution as a whole? And particularly when the particle size matter of 2.5 comes into the picture and you inhale that normally as we normal breathe. So that are the major things which actually lead to quite the deleterious conditions and the underlying respiratory symptoms. This is a photo, I think it's 2019, but it happened every year. We are currently in Australia in summers at the moment. But the 2019, the image which is on the screen was the catastrophic threat. And the reason was because there was a lot of loss of the wildlife, and a lot of pollution has come through across Australia just because of the burning of the bushfires. And the burning of the bushfires was just happened because of the heat. And I'm not showing the the burning of the forest. What I'm showing you here is the kind of the smoke we generate with that and people inhale. Imagine a person with a lung cancer, or imagine a person, an elderly person, or imagine a person, a pregnant woman, which has to go out in this condition. What the person is inhaling is not a right thing. That can further lead to quite a significant deleterious effects on their body and on their health. Now, if you look at this image particularly, this is Sydney, and this is the Sydney Harbour Bridge, and this is the center of the city. This picture was clicked by one of my own colleague while passing through the Sydney Harbour Bridge. So she was just need to enter into a Sydney Harbour Bridge from here, and you can clearly see this is smoke. And this smoke, despite the sky is most of the time very blue, crystal clear, always most of the time. But this is a time the smoke has even come to the cities. So not only just it's affecting the country people or the rural people, it is even affecting the cities as well. It's not only restricted to Australia. In Canada as well, there is a significant loss of the uh, mortality just happened because of the smoke cases and stuff. And so as it happened in United States as well. It also happened in Indonesia when they burn their forest, and the smoke goes to the Singapore and Malaysia. So it is actually a problem across the globe if we really look. So now I'm going to show you actually a bit of my research. Can I just have a 2 minutes break? I'll be back. Sorry. I'm sorry about the disturbance. I thought there is something downstairs. Sorry. So in my team, actually, I worked on both phytoceuticals and nucleic acid-based therapies. Now, I work significantly on nucleic acid-based therapies as well, particularly microRNA inhibitors, decoy oligonucleotides. And I do that because not to run behind the race. People talk a lot about mRNA vaccines and other things, but phytoceuticals have its own unique aura. And this is what I'm going to talk today, actually, being very passionate about these. These are some of the drugs I believe everyone knows. Curcumin is the gold of India. Everyone use that, excuse me, everyone use that. We have also started our studies with curcumin, and we have done so many studies at the moment with different drug compounds, starting with berberine, rutin, agarwood, chrysin, xanthomol. There's plenty of drugs we have worked on. So, and today my talk is all going to be focused on phytoceuticals. I have a lot of data on a nucleic acid-based therapies, which my team do, but I'm not touching on that today. And all talking in terms of the lung health. So with the agarwood, even one of our patent, our PCT has just came 2 days, well, last week actually. So, which is the agarwood oil nano emulsion, which we are using it for the lung cancer. It is a project of one of the company which is based in Melbourne. Now, in respiratory diseases, what exactly happen? So when anyone get any of the chronic respiratory disease, whether it's asthma, COPD, any, they are major features of any respiratory diseases are airway inflammation, and people have difficulty in breathing because the airway become really narrow, so the no exchange of the gases happen. There is increase in the airway hyperresponsiveness, and also remodeling happen where epithelial cells goes into the transitioning and changes, and a lot of mucus production happen, and that can lead to the chronic respiratory diseases. You go to any clinic, respiratory clinic, or any doctor around the globe, they give you these two things. They give you inhaled and systematic corticosteroids and beta adrenergic agonists. These are the two things which they give you. And particularly when people are continuously taking corticosteroids for a very, very long time, a threshold comes when it stops actually reacting or behaving. And that's what we call as a steroid insensitivity. And that when it stops actually showing their therapeutic effects, then what's the treatment? There is no other treatment. At the moment around the globe, the most commonly used steroid is fluticasone. But that will also show a steroid insensitivity at some point of time. After that, there is no treatment. Then where to go? So there is what we need, some new therapeutic interventions. And I can bet on the screen, and the people, those who are attending this conference, I can sure that in your family or you, definitely someone had some kind of the underlying respiratory condition, whether it's asthma or anything. So, and I can also tell you there are almost around quite a significant percentage of asthma which is remains undiagnosed, because people even have asthma and they don't know they have asthma. So that all leads to the need of the urgent innovation therapies. Now, why plant-based therapies? Where people, when I these kind of the presentation across the Western world, um, people do ask me why you want to work on a plant-based therapies. Despite you won't believe, the Western society is significantly actually absorbing and applying our culture from the Asian culture, whether it is yoga, whether it is holistic approach, whether it is complementary and alternative medicine. If you go in a chemist shop here, you will see the first 10 shelf full of the complementary and alternative medicine. And they contain half of the drugs which I have worked on. but they all comes in a capsule or a tablet form, and the drug might be in that maybe up to 1000 milligram or 500 milligram, but where the absorption happen? No. Until and unless we're not going to use some advanced delivery, the absorption is so poor because most of these plant-based drugs are water insoluble. Dr. Daya have already talked about enhancing the solubility and stuff, which is so true, and they can be even applied to these phytoceuticals as well. And when they talk about the Western world, the holistic approach is known for ages, and particularly look at this image, which clearly depicts in one of their own that plant-based medicine in the 17th century in Britain, they were been using it. This is the real graph, which clearly demonstrate why there is a thirst or a drive towards the complementary and alternative medicine and plant-based medicine. Till 2029, you can clearly see how the graph is rising across different countries with the investment in the amount which is being going to be used for the global health medicinal products. So you can clearly make out that how impactful it is in the coming time. And this is just a distribution across the different formulations which people use, tablet, capsule, powder, extract, syrups, which are commonly used. Even in Ayurveda, it is being commonly used at that time. So that clearly shows that why the plant-based medicine are very much needed. Now, if I particularly highlight this news, which happened actually in 2022, not very long ago, just now, you can clearly see actually that Ayush, which is the ministry, which is now had so many grants which they've been giving across to the people. They had even signed one of their MOU with the WHO to have a center of the traditional medicine. And if you really look at that, the WHO also have established actually Global Center for the Traditional Medicine in India, and it has been completely evolving up to the dollars of around 3.5 million, which they have been invested around that. So it is completely rising, and I'm not making this. This is all on the news. So whether it is the revenue generation from the phytoceuticals or whether it is a research in that area, it is quite clear. So coming to my own research, and let me show you some of the research which we've been doing. This is one of the very old research. When we started, as usual, everyone start with curcumin, and we developed the liposomes containing curcumin. So these are the two students from Malaysia. They were the visiting student, Jinjing and Elaine. They actually, one student developed liposomes, other developed the niosomes, and we tested it on the human bronchial epithelial cells. They're the real human cells which are obtained from the patients. And when we treated our liposomes, I'm not showing any kind of the formulation data. I'm just showing more of the biology and the interventions and the results. It was very obvious and very clearly it has shown that all the hallmark features like IL-6, IL-8, IL-1 beta, and TNF-alpha was significantly reduced, which was all the major hallmark features that are involved in the pathophysiology, particularly inflammation and remodeling. We also developed actually niosomes as well, and they have also shown a similar kind of a trend, what it has shown with the previous liposomes as well, in terms of reducing all the inflammatory markers. And particularly, you can clearly see that we have compared this with the marketed product, which is the salbutamol here, and now we normally use fluticasone, which is the standard corticosteroids commonly employed. This is one of my industry project from the Pharmacobiotechnologies. It's a Sydney-based company. And they had developed liposomes containing curcumin. They named it as a plexosome. You can Google it. They're very widely well known. They've been selling them as a nutraceuticals quite significantly. This is some of the people of my research team. Sophia is one of my master students. She's just starting her PhD. This is her project actually. And Gabriel finished PhD with me, and he was contributing to this project. Keshav is a senior postdoc, and Manisha is a visiting researcher. They all contributed to this project, and we have clearly shown that when the curcumin liposomes being treated with the cancerous cells of the A549, it had actually two important pathways, the cell survival and the invasion or the metastasis through the APCAM, estrogen receptor alpha, they've been completely reduced. We have recently also published in Halion as well. Oh, sorry, it's there. I'll show you some of the other studies. So we have also shown not only in lung cancer, but we have also shown in the cigarette smoke extract, means according to the COPD, it also actually reduces the hallmark features of like IL-8 and IL-24. And you can also see this is a continuation of the cigarette smoke study, where we have shown that it also suppresses senescence. Senescence is nothing but it is more of a cell aging. And you can see, referring to the paper, there are a lot of proteins which were relating to the inflammation and aging, which were actually being suppressed by these liposomes. And this is our very latest paper in continuation to the this study, which I was showing you, the cancer study. This is a very detailed study in which we have shown the proliferation, migration, and apoptosis, all the important hallmark feature of the cancers, are completely been suppressed by using the curcumin liposomes, or we call plexosomes. This is another project of D. Aurora, where I just mentioned in the beginning. This is a company which is a cosmetic-based company, and they do a lot of cosmetic products using agarwood oil. And agarwood oil is a very well-known compound which is used in the Middle Eastern countries, particularly for the sleep disorders. They gave that oil to us and we developed this nanoemulsion. We have tested this against inflammation. We have tested this against the cigarette smoke COPD, and we have also tested this against even the lung cancer. The lung cancer, I'm not showing the data because this has just gone into the patent. The PCT has just released 2 weeks ago, and it is online. The studies are now, PCT is online, so it's all well protected. We just in a process of, the company is in a process of getting this across the across the patents in different countries. So this study we done actually with the cigarette smoke induced inflammation and oxidative stress in the airway epithelial cells. Ronan is a senior research director and on a board of directors now actually with Aerogen. Aerogen is a topmost research, inhalational and respiratory based company in Ireland. He actually contributed significantly on this project. So when we have treated the cigarette smoke on these cells, and after treating our nanoemulsion, we can clearly see that a lot of anti-inflammatory cytokines and mediators and pro-inflammatory cytokine and mediators, they were suppressed, but anti-inflammatory mediators and cytokines were quite predominantly upregulated. There were certain antioxidant genes and the pro-survival pathways we looked at, particularly down the track. PR3 kinase is the one which was highly picked up. The anti-cancer potential is not under patent. It is already been actually now already in the PCT. This is another company, and they have just given me another set of funding yesterday for next 4 years actually to further their research. This is an Indo-German biotech company called Medicinals. It is based in Pune and in Germany. So Joachim is the German counterpart and Prakash is the Indian counterpart. They actually developed this medicinal during the COVID. And all the information is on their website. It's freely available resource, and you can easily see it. It contains 9 different phytoceuticals in a suspension form, and it has proven, they've done animal studies, human study, everything, and it has proven quite effectively therapeutically effective in COVID-19 and long COVID. They gave their formulation to me and I tested it against a bit of the lung cancer, and it can clearly show that tumor suppression, all the angiogenesis inhibition, proliferation and migration related factors were quite significantly altered in terms of reducing the pathophysiology of the disease. This study was carried out by my research assistant, mostly by Rashi, along with closely working with our senior postdoc, Keshav. This is another study with the three of the Malaysian students, which came actually almost in 2020 in my lab, Li Hian, Chianming, and Hu. And so they actually used naragenin, and they have also shown that all the anti-proliferative activity and all the important cytokines were significantly reduced in terms of the anti-inflammatory and the cancer study I can show you in a minute, when actually treated with the naragenin containing liquid crystalline nanoparticles. This is another group of the students which also came through Malaysia to work in my lab with Alan from HMRI, and they worked on quercetin. They done actually two different studies. What they have shown is significantly there were features of the anti-inflammatory IL-6, IL-8, and IL-1 beta were significantly reduced. And you can clearly see here the treatment, the standard was fluticasone, because it is like a 2 years or year and a half ago, this study was published. The same group also worked alongside the, you can see that they're not only just on the quercetin, celestrol, and celestrol also have shown the similar features, but it didn't pick IL-6, IL-8 or anyone. It was only the IL-1 beta.
नहीं कंप्लीट कर दी उसने क्या रेट दिया है
सर उसमें रेट बहुत ज्यादा है 1 लाख समथिंग है वो टोटल फीस है तो वो लेना ही नहीं बस कंपैरिजन के लिए रखते हैं
ठीक है
सबसे अच्छा तो ये हो गया हमारा 30,000 छोटे दिला के सब निकल गया ये बहुत अच्छा हो गया सर ये निकलना सर 50
Excuse me, sir. Participants, kindly mute your mics.
कुछ जनरल सर मंगाने होंगे क्या आप ये कर दिए कुछ मैगजीन्स जनरल
Okay, so the other important study we've done is with berberine, and this study has really this study ours is actually gone quite into the public domain.
Sorry, sir, the inconvenience, sir. Just a second. Participants from SMS College of Pharmacy, kindly mute your mic.
Okay, so this is one of our another study on berberine nanoparticles. For berberine, we use two different kind of polymers. We use phytenterol and monolin, and we done the liquid crystalline nanoparticles. And we have clearly seen actually there's significant pathways which has been triggered in the lung cancer. They've been through proliferation, apoptosis, migration, and even through the epithelial mesenchymal transition as well. We have continued this study in different, not only just restricting to cancer, but towards the normal human bronchial epithelial cells. And they have also shown CXCL8, CCL20, and HO1 are the one which were being quite down-regulated with these particular treatments. So Vamshi was my PhD student. He finished his PhD and he was involved in this project. He had even significantly worked on the epithelial mesenchymal transition. This was the phytenterol study. This was done by Vikas, my research assistant, and as you can clearly see, there were a lot of proteins which were quite significantly down-regulated with the treatment. And also in the cigarette smoke, the study which we done with Amlan from Manchester, and it has also shown that it reduces actually the oxidative stress, senescence, and inflammation when those berberine nanoparticles were being treated. And it actually had significantly, not only just through the COPD related proteins or inflammatory genes, it has also, you can see there's quite a number and bunch of the cells which were actually being inhibited. And that clearly showed that all the pathophysiology of the COPD was significantly reduced when treatment with those. So when this study was published, actually SBS is quite a wide known news channel. They actually interviewed me. After they had interviewed me and they published this, actually, this news was almost, and these papers were almost been published more than 25 countries in different channels, in different languages. So you can clearly see, because berberine is a quite commonly used traditional medicine even in China. These are just some of the glimpses where it has been completely gone wild in different channels, including the German pharmacy board has even done on their pharmacy board portal as well. This is one of our latest study in 2024. We have started using some of the drugs for the repurposing, and ribavirin was one of the drug which we have significantly found it is quite effective in lung cancer. And we have shown this through the biological experiments as well as through a detailed computational.
Calculator.
So through a detailed, actually computational studies, we have shown the relevance of the same predictor markers as compared to our identified studies. We also been working on microbiome extract as well, and also been showing the how it can affect the remodeling and the lung cancer pathogenesis as well. We have recently also been working on a new drug called Fistine, and we have also developed nanoemulsion, which has also shown quite effective in lung cancer. This is a very old study, but this is all about the animal studies where we talk about when any patient or any person have a COPD and they get flu on top of that, which is the influenza infection. It actually increases the level of microRNA levels like microRNA 125A and B. And in one of the APP previous conferences, I presented this study in detail with all the data in there. So we have shown that through the A20 and MABs, it got downregulated with the increase in the NF-kappa B level. And when we treat actually the, and that ultimately increases the influx of the inflammatory cytokine. But when we give the inhibitors of these microRNAs, and currently we are already have done some nanoparticles containing these inhibitors, and we have done some studies, it is clearly showing it is actually reducing the pathophysiology of the disease. We also work on asthma. So in asthma, we normally use the ovalbumin method. We actually infect the mice with flu, and then we give the ovalbumin to develop the asthma over the period of time. And then what we have seen is when the allergic mice with the infection always had a high viral titer and also quite a high significant number of the inflammatory cells, there are a lot of interferons which are needed to fight the immunity and to bring the immunity are quite downregulated. You can clearly see IFN beta and IFN lambda, they are quite significantly downregulated. Airway hyperresponsiveness was significantly high in the mice, which is having both the allergy and the flu as compared to allergy and flu alone. Asthma, normally the eosinophil cells got quite high, and that's what we observed in our model. And when we looked at the IL-13 level, that was also very high, along with the respective IL-13 alpha receptor, and also the mucus production around the airways was significantly high in allergic infected mice as compared to allergy alone. Then we gave, because we have observed here that IL-13 was one of the predominant factor which was actually causing the allergy, then we gave anti-IL-13. This was being made and developed through one of our UK collaborators, and we can see there is a lot of features which were significantly reduced. There were reduction in the airway hyperresponsiveness, viral titer, eosinophils, and mucus as well. So what I have shown you is a blend of quite studies of the in vitro and in vivo. And I do get a lot of emails from real patients, particularly for the berberine one. Is it your treatment is in the market? Because that has gone really wild in the news. So we are currently working towards the translational pathway with some of the industries actually seeing if this can really come as a cure for lung cancer, which is still need to do a lot of clinical trials and other things, and you need funding for that. I've been applying multiple grants, but been highly unsuccessful, not successful. Most of the funding which I have is from the industries and from the other small funding bodies, but the major fundings which I'm applying up to $1 million, I've been mostly knocked down with very less scores. Means with the cutoff of, like, if the cutoff is 5.4, I'm sitting on 5.3 out of 7. So I think this is a real research trend across the globe in terms of funding, but I hope that there should be a miracle happen where people can, really see the extent of the research we're doing and why to help the community. And you can clearly, actually, looking at these emails, you can clearly see how people are actually been talking, whether we can take those berberine, nanoparticles. But the problem here is I usually advise you can talk to your clinician. There are a lot of berberine based nutraceutical and phytoceutical formulations exist in the market. So you can use that. However, you used under the supervision and guidance of the clinician. I think that ends my talk. And before I end, actually, I would like to really acknowledge the Woolcock Institute is coming on the top first. That was not the intention. I'm a part of a Woolcock Institute, which is the topmost respiratory institute in Australia. Half of my team sits there, half of the team I work in UTS. I would really like to thank the organizing committee of the Associate of Pharmacy Professionals, 13 annual international convention and the 7th Indo-Australian conference. And I'm really proud to be leading the Indo-Australian chapter of this APP. And thanks to actually Dr. Rajiv Dahiya for being, giving me this kind of the opportunities. Particularly, it's almost 13 years. I can't even believe how the time flies and we get old, particularly an association also getting a bit of the, aura in terms of growing. So it's congratulations on that. Thanks to Dr. Rajiv Dahiya and Sunita Dahiya. And also the school where I come from, the Graduate School of Health. Charlie was the founding dean. Kylie is the current head of the pharmacy. I'm also a part of the Australian Research Institute of Complementary and Alternative Medicine, which is called ARCM. And I'm the lead of the drug delivery node or translational, where John Adam, he's a distinguished professor. He's the lead there. And Brian Oliver is also a distinguished professor who works very closely with me. He's from the lead of the Woolcock Institute, where I have my own team as well, and he's a part of School of Science at UTS. The most important people actually who have generated all the data which I have presented today is my team. So many of people have graduated, many of people are still there, and it's been in and out kind of a people. They are the those who've been working hard and actually generating that data for me. And a lot of funding bodies, those who've been actually putting their money in to do this research, and my industry collaborators. So I think with this, thank you so much, everyone, for being patient and listening to me. And I'm happy to take if you got any questions for me. Let me stop sharing now.
Thank you, Dr. Dua, for your informative lecture. And I over to the judges if they have any question.
Good morning, everyone, sir. Sir, myself, I'm Rohini from VIPW studying 4th B Pharmacy. First of all, it's really fortunate enough to hear from this morning with your very much elaborative work on phytochemicals related to respiratory based problems. So here my question is, from our theoretical studies, we learnt of plants based on chemical constituents, but going to research, if we select any new plant, how can we identify any important constituent that is responsible for pharmacological activity, as we have so many constituents in any part of the plant?
Yeah, so I think, look, I'm going to give you a real answer. I don't know. And the real answer is because I am not a pharmacognosy person. So it is an area which sits with more of the pharmacognosy people, which actually do the extraction and also actually do the isolation of the pure compounds. In whatever the studies I have shown to you, they all are being the pure compounds which we got from industries or either we have purchased commercially available, which are in the market. So, but however, just to add on, like how it can be done, definitely more of the various analytical methods, particularly nowadays, the MS and other methods are being commonly used where you can have a detailed isolation of the compounds, but every single isolate of the compound need to be tested through the biological effects. Then only you can say that this compound is the right compound which is showing a potent activity and this compound is one which is not showing. Even you think that the results what I have shown you is a study which has been done in the last 8 years. So we got so many negative results sometimes, so many negative results. I'll give you one recent example. One of my student, Adam, he's a PhD student, and he came actually, he's working on A509 cells actually. And he came with me showing that the 15 nanoparticles, what we have developed, it's not showing any anti-cancer activity while it's showing the results other way around. It is increasing. It is making the cells to grow faster. So when we were doing the scratch wound healing assay or proliferation or migration, nothing. It was just growing the cells. And you won't believe, we later found that in our formulation, we have a glucose or dextran significantly high. And that is a source of energy for those cells. So what I'm trying to say here is it depends on the deeper investigations on the pharmacognostic side, and that can actually identify which compounds you need to go with and which compounds you need not have to go with. But sometimes hit and trial work, sometimes hit and trial even don't work.
Thank you, sir.
No problem. Thank you.
Good morning, sir. I'm M. Harshita from Vijay Institute of Pharmaceutical Sciences. Sir, as you said, liposomal curcumin prevents epithelial cell damage in cigarette smoking induced epithelial cell damage, sir. So I'd like to know, are there any products in the market which are having liposomal curcumin?
It is plexosome. So plexosome, which I showed on my screen, and the one I tested on the formulation is a marketed drug. It comes in a box. It's a plexosome is in the market. But I'm sure there are so many curcumin formulations, but I don't know.
ఆన్లైన్ అనుకుంటా ఇవాళ
Good morning, sir.
Yes.
Myself, G. Krupamayi, Assistant Professor, VIPW. Sir, here today you have emphasized on very novel therapeutic interventions for chronic airway diseases by using phytochemicals, nucleic acid-based therapies. And also you have told that India or many other countries were suffering from smoke or any fog. Sir, we heard that there are a few pollen grains related to trees like Madagascar jasmine, S. floribunda, Conocarpus. All these types of plants and trees are also releasing some allergens into the nature. So can we identify these respiratory problems which are based on which type of allergens or which type of problems, and can we prescribe the suitable formulation for that patient?
Yeah, it's a very good question, actually. So probably you might have seen, even if we get any allergy to anything, we usually go for the allergic test, right? When we do our allergic test, then they say you are, like my daughter, she is allergic to lactose. So no, sorry, she's allergic to gluten. So she can't take any bread. She even can't eat, chapati or anything. So we give her lactose milk. So if she gets that, then she gets a lot of body, like rashes. And I'm allergic to mushroom. So if I eat mushroom, I get a lot of rashes on my body. In the same way, exactly, those particular pollens, even in Australia, Melbourne had a cytokine pollen storm. And it came through where? God knows. And that had led to the cases of almost 350 people in 10 days. Despite it's a very small country. So definitely people are working on that. And particularly nowadays, the single cell sequencing is the one which they are using, which I even came across the paper yesterday. One of the single cell sequencing is one of the studies which they are doing it to identify certain time of the allergens. They're not saying allergens, they're using the term insult. Insult can be allergen, it can be pollen, it can be smoke or anything where their chances of the reactions of the airway hyperresponse and other things are quite high. So yes, it's happening.
Yes, sir. Thank you.
No problem.
Good morning, sir. I'm J. Sunita Blessu of PharmD 4th year, studying in Vijay Institute of Pharmaceutical Sciences for Women. So my doubt is, as you said, there is the preparation of nano emulsions, no, sir. So what were the challenges that were faced in developing them, sir?
I think a formulation person, any person, even your teachers can tell a pharmaceutics person. So whatever a normal emulsion, you might have prepared an emulsion in your undergrad program in the lab, right? And where when you have done that, you might have seen the most important part where you can mix the oil and water, and then there is a chances it may break. That might not bring the stability. But what we are talking here is the drug need to be stable over a longer duration of time, and it should remain intact so that patient can have a homogeneous, patient compliant dose. So when we developed our batches, and I think Dr. Sunita Daya also showed some really good case studies where the optimization she showed, like on different controlling the parameters. In the same way, when we develop those formulations, it's a hit and trial, changing the percentage of the emulsifiers, and those kind of things actually can help you identifying the problems and also developing a suitable, stable formulation.
Oh, thank you, sir.
No problem. All right. Thank you, everyone. Thanks, Dr. Daya.
Yes, thank you, Dr. Dua. We can move over to our next expert, Dr. Batri Selvan from Sunway University, Malaysia. and I'll ask the co-convener to go through his profile, please.
Yes, sir. I invite Mr. S. G. Bhavani, Assistant Professor, Department of Pharmaceutical Analysis, to introduce the profile of our keynote speaker, Professor Vetriselvan Subramanian.
Good morning, everyone. Myself, G. Bhavani, Assistant Professor, Department of Pharmaceutical Analysis, VIPW. I feel honored and privileged to present the profile of Vetriselvan Subramanian, PhD, FMSc. Professor Vetriselvan Subramanian sir is currently working as Professor of Pharmacology at the Department of Medical Sciences, Sunway University. Sir was graduated from Annamalai University and began his academic journey in the year 2008 at Rajiv Gandhi University of Health Sciences, Bangalore. Shah has more than 15 years of vast experience in his career, accumulated to teaching and research experience at various prestigious institutions like Arba Minch University in Ethiopia, Masa University in Malaysia, among others. Professor Vetriselvan Sir has held prominent academic leadership roles, including serving as Dean, Deputy Dean at Masa University, and held visiting and adjunct professorships at institutions in China, Cambodia, and India. His supervisory mark is extensive with 28 completed students' work, research projects, and ongoing PhD supervisor at multiple international universities. As a prolific researcher, Shah has authored over 250 papers with a cumulative impact factor exceeding 700. Shah has filed 8 patents internationally and contributed to 6 book chapters. Shah's expertise is widely recognized as evidenced by numerous grants including FRGS grant from Malaysia's Ministry of Higher Education. Shah is an active member of several professional organizations, including the British Pharmacological Society, the Indian Pharmacological Society, the Obesity Society, and the Malaysian Scientific Association. Shah's research mainly focuses on advancing drug development and pharmacology. Shah has delivered keynote addresses at over 25 conferences worldwide. With this brief introduction, I extend my sincere gratitude to Professor Vetriselvan Subramanian sir for spending valuable time and sharing sir's expertise and insights to us. Thank you, sir. Thank you for giving this opportunity. And now I invite Professor Vetriselvan Subramanian sir to deliver his talk.
So, thank you very much for a wonderful introduction on my profile. My sincere thanks to Raju Daya, APP President, and Sunita Daya, and the President of the Principals and President of the University. So I'm very happy to be here to share my experience and to share the research interests and how the research translate to the next level. Mainly, I'm going to focus on translational research. So with this, my sincere thanks to all the participants and organizing committee members. So this is my. Can you see my slides?
Right now, we are not able to see, sir.
Okay.
But
Can you see my slides?
yes, sir, we can see.
Okay. Once again, thank you for everyone. So my today topic is a translational research, bridging is science and practice, mainly for how the lab research translating to the clinical studies. So, as I mentioned, my thanks to APP and Vijaya Institute of Pharmaceutical Sciences in Andhra Pradesh. I'm working as a professor of pharmacology at Sunway Medical School, Malaysia. This is my school, which is located in Kuala Lumpur, which is the major city in Malaysia. So one of the topmost in private university in Malaysia is a top one university. We have associated with many overseas, it means foreign universities, including University of Cambridge, University of Oxford, and other universities. We have a own medical center and a biomedical research center at Cambridge University. Recently, in 2023, I obtained a few grants from Ministry of Higher Education. We used to call FRGS, a Fundamental Research Grant Scheme. Also, I obtained two more research grants from Monash University, Australia, and UKM local universities and national grants. So far, I have supervised more than 25 students. Also, currently, I am supervising with many universities, including India and Malaysia. Especially in University Malaya is one of the topmost university in Malaya. It's a number one public university. Also, I have associated with Monash University, Malaysia and Australia. So why the translational research is important? No, so how this research is translate or apply for clinical knowledge or practice for the patients. So which is very important. Lab research, especially we do only with the basic research, the chemical test or in vitro studies, translating to the preclinical studies, animal modeling. So when we do with these kind of studies, should be translate to the clinical applications. This is we used to call translational approach. So when what are the objectives of clinical studies or translational research? Participants will effectively translate lab discoveries into the clinical applications. So this is the meaningful research. It's not done by for your research projects or for your only the publications. Also should be translated to the next level. Learners will design and execute is a translational research ensuring ethical and safety and compliant studies that successfully translate scientific discoveries. Also, graduates will communicate complex scientific concepts to discover and diverse audiences. Different group of people have to be, explore the knowledge on your research, articulating the significance of translational research findings for healthcare professionals, policymakers, and public. So these are the main objectives why translational research is important. So translational research refers to applying findings from basic science research to enhance the human health research. So it involves translating knowledge from the lab research to clinical practice and community settings. There are 10 key points which are very important for translational research. I am not going to the specific model or animal modeling or specific disease conditions. Just generally, I am going to explain why the lab research is important. So lab research is not only for identification of molecules or molecular mechanism, identification of molecular mechanisms or identification of in vitro cell line inhibitory responses. No, we should translate to the next level. Should be whatever research we done at the lab research should be translated to the clinical approach. So the translational research begins with identifying fundamental scientific principles and discoveries in the laboratories. It could involve understanding molecular process, genetic factors, or physiological mechanism. Why we wanted to go for lab research? We need to understand the chemical compound. What kind of chemical compound, whether it's a toxic or safe. So then we need to know whether really have any toxicity or is there any therapeutic response, so that we need to go for some test or some parameters to identify the pharmacological responses on the test compounds. Whatever test compounds, even though the synthetic compounds or natural compounds, any compounds should be have solid evidence. So with the basic evidence only, we need to go for further test, or maybe from the preclinical to the clinical research. So this is the basic science. Why is a how is important. Is a really is important. Without understanding of fundamentals, we cannot go any advanced research. So the basic research, which is very important. So preclinical research, once it means once we have done the cell culture work, or maybe in the chemical test, then we can go for animal modeling. Especially why we need to go for animal modeling, then only we can understand the physiology, mechanism of physiology, and mechanism of internal body responses. We cannot go simply after in vitro test, cannot go into the clinical research. We should have solid evidences at the different levels of research, including lab-based research, basic research like a chemical test or in vitro test, animal test. Then only we can move on to the clinical design or new drug discovery. It will take a longer time. It's not a short period of time. It will take more than 15 years also, some kind of research discoveries. After 1978, there is no much antibiotics like antimicrobial never coming out to the market. It's due to the lack of research studies. There are many gaps because funding is one of the main factors. It's very expensive. So there are a lot of difficulties are there. But nowadays they move on to the is a replacement therapy, immunomodulatory therapy is a because the immunomodulatory therapy sometimes we reduce the cost. These kind of new approaches are ongoing currently because to reduce the expenses. So identification of targets. Any researchers, when they identify any molecule without base reference, they cannot move on to the any lab research also, because they should know any molecule. They should know some basic evidence like reduce the oxidative stress, sometimes to reduce the inflammatory markers, to reduce the body cyclooxygenase enzymes. So certain level of evidence they should have, then only they can go for some studies. Let's suppose, let's say if there is no evidence, but ancient times they're using continuously, since ancient times we are using some kind of phyto compounds, but there is no evidence. Definitely we can move on to the basic research to identify the pharmacological screening. So what effects on the particular compounds. But whatever we go for any advanced research, we must have solid evidence and also appropriate guidelines we have to follow. Without following any guidelines, our drugs does not accept by the any authorized board like Food and Drug Administration or local food corporations. They never accept our research work. So we should have proper evidence. So once we have completed the lab studies, then you all aware of this. We move on to the clinical trials with a small group of populations with the minimum 20, maximum is 100 participants to identify the safety and efficacy of the drugs, mainly to identify the safety and toxicity. So whether sometimes in the animal model have a high efficacy, but when you go for clinical trials, there is no effect, failure. So we cannot move on to the next level. So we have to stop. So likewise, there are 4 steps we are going to have in clinical trials. The first, the phase 1 with the minimum participants. So mainly we investigate on the safety and toxicity. Followed by, we can go for phase 2 trials. Mainly, we identify the mechanism of drugs, how the drug respond to the patients. There are many factors involved, individual variations. Some patients may get a therapeutic response. Some patients may not get any response. Depends on the individual's genetic factors. So many factors involved to produce the response. Also, this kind of study is very important to find out the therapeutic efficacy or to give the concrete report whether the drug have safety or any therapeutic mechanism or not. So the phase 2 is a maximum we can take 300 participants. volunteers with their consent. Without consent, we cannot take any patients for clinical trials. So this study, which is very important to move on to the, is getting approval. Then followed by, when we move on to the phase 3, maximum number of patients, participants, around 3,000 participants. So this different ethnicity and different group of people we are going to involve in this study. Why? Because diverse people we must involve. Then only we can identify the response. Some group of people may have a high response. Some patients may not have therapeutic response. Some patients there is no effect. So we need to conclude majority of responses and minimum responses. Based on that, we can go for a report and getting approval process. Then followed by phase 4 is mainly for regulatory approval, which is mainly we are going to get the approval process based on the preclinical studies, and based on the clinical studies, we are going for approval for the drugs, the new drugs. So this one we need to submit all the data with the evidence. The data should be appropriate. Also, we have to submit each and every steps and approval process with the evidences. Then they can go for necessary approval process. Or is there any queries or questions? Maybe they can communicate with the respective researchers and research centers. So this is the phase 4 studies. So why implementation in clinical practice? So when everything is over, example from the lab research to the clinical trials, then we are going to implement into the clinical practice, apply to the patients. This is the most important and crucial, is a stage. Sometimes after post-marketing also there are some issues. When we use largely like a large group of populations is with 100,000 people or 200,000 people, sometimes we may found we may find some kind of adverse effects or side effects immediately. If there are a lot of reports coming, it means by the clinical experience, then immediately we have to stop the medication. It means they withdraw from the market also. This is based on by the patient clinical experiences. The patient experiences, we continue the market. Even though some of the drugs nowadays in the US, many of more than 15 years we are using some anti-inflammatory drugs like NSAIDs. But recently they withdrawal from the market. Example for nimesulide. There are many drugs with already withdraw from the market. This is based on the patient experience. So likewise, so when we go for any new drug implementations, there are some risk. We have observation period, minimum 1 to 3 years. They go for close observation from the different countries, people, how they're getting response. Is there any major side effects or any adverse events? Immediately they stop from the market. Also, health policy and guidelines, whatever we go for any research, we even bench research to bedside research, we need to follow the guidelines, proper guidelines to provide proper guidelines. Based on that, we need to give some advice to the healthcare providers and policymakers to go for further decisions. So, which is very important to implement and to execute our research and the final products. Monitoring and evaluation, continuous monitoring, which is very important. Evaluate the practice, the clinical experiences, and when the doctors prescribing the medications, after taking the patients, how they feel. Really, they feel good. Minimum adverse effects with the good therapeutic responses always acceptable. If there are many side effects, immediately we have to take a necessary action to control the unnecessary side effects, which is very, very important. Importance of translational research, which is you all know very well. Nowadays, many people are talking about translational research. What is a translational research? From the basic research to clinical research. So whatever research we are doing, everything is a translational research. But that's what, no, this is the reasons. Whenever we do any kind of research, we should have the future or future directions. Why every time they're asking in the exams or the PhD students during Viva sessions or master students during Viva sessions, final presentations, they're asking, oh, what is the future directions, the future ideas? Your research should be translate to the further. It means translate to the clinical applications. It's not stopping and only within your master degree or PhD level. So should translate to the next level. So these all research we used to call translational research only. But translational research is accelerates the transformation of scientific discoveries into the practical applications to makes and to reduce the time taken process for innovation to reach the patients. It means patient clinical approach. So which is very important, and also improved patient outcomes. When we go with the connective research pathways, we can easily address the gaps. There is no gaps, so we can have a proper evidences. So finally we can go for the proper therapeutic applications based drug developments. So these all which is very important to focus during our universities research work. This one easily we can translate to the clinical applications. So these are the points we have to take a note on when we go for any kind of research. Also, optimizing healthcare resources. Also, within a pharmacist or within the medical doctors cannot do anything. We must have a good collaborations, and also we must have other researchers. Healthcare professional support, including nurses, including lab technicians, lab scientists. We must have connected with all the researchers. Then only we can get a lot of ideas to enriching the research activities. Also to find out the research, new insights to develop the novel drugs. Within the pharmacist cannot, because the pharmacist may not know the clinical experience, real clinical scenarios. We must have an interaction with the medical doctors or physicians or oncologists, or it depends on the research work. We must have this kind of collaborations and collaborative workshop. Then only we can get a lot of ideas to go for translational research. So many people are involved during translational research: public health expertise, basic scientists, preclinical scientists like clinical scientists, and other healthcare professionals. So then only we can develop the novel and is a newer drugs. So importance of translational research is a very important. Translational research allows the development of personalized and targeted therapy. So sometimes, let's say example, there are two patients, cancer patients, same cancer, like a colon cancer. When we are treating the patients, some patients get very good. One patient get is a good therapeutic response, better therapeutic response. Another patient, there is no therapeutic response. Why? Because there are a lot of factors are involved, like genetic factors, genetic linkages, and also the patient differences. There are many factors involved within the body system, so that the depends on the patient conditions, depends on the patient body systems, or based on his comorbid. We have to go for personalized treatment. It means based on the patient conditions. We cannot go for one treatment for every patients. The one treatment approach for all patient is not possible. Sometimes we need to look into the patient conditions and internal body acceptance of the drugs. These all, there are many factors are involved during the therapeutic approach. So this is we used to call a tailored therapeutic approach, so which is very important. And also the health practices, clinical practices, which are very important. Always we need to have precautions on health protection, health services, and health improvements. This research, particularly the translational research, contribute to the evidence-based public health practices and policies leading to more effective prevention and management of disease at the population level. So each and every process or each and every research stages, we should have proper evidence and guideline we should follow. Then only we can give clear idea to the policymakers and healthcare providers. And also interdisciplinary collaborations, which is very important. Just now I mentioned, within pharmacists, we cannot do everything. We must have collaboration with the clinicians, policymakers. They give a lot of idea on patient perspectives. Also, the stakeholders also give the business aspect. Also, whatever you develop any newer drugs, should apply for all group of people, not only for rich people, not only for poor people, irrespective of any economical boundaries. Your drug should be useful for everyone. So the multidisciplinary collaborations and approach is very important to get more ideas and their thoughts and their inputs to go for research work. It means to go for next level. Economic impact is very important. Successful translation research can have significant economic benefit by spurring innovation, creating jobs, and reducing the economic burden of diseases through improved health outcomes. So the translation research we used to call evidence-based research or continuous research or connecting research. This research is very important to address the gaps and to reduce the global economic burdens, also to reduce the burden of diseases. So these all important aspects in the concern of economical aspect. Also demonstrating the translational research potential of research findings importantly for supporting discovery and innovation, advancing knowledge and understanding, driving economic growth, promoting collaborations and networking, addressing societal challenges. So there are many lot of advantages when we go with the procedure of translational research. Also the translational research, which is there are many goals why we need to go for translational research. The main reason is should be apply or should useful for the public health practice. It's not within the lab studies or lab report. This research should translate to the next level or real world problem solving. So whenever you do any kind of research, should move on to the next level. Then only we can treat the patient, we can apply at least the minimum level of clinical practice. With this, addressing unmet medical needs. Why the translation research? There are a lot of research so far have done, but none of the drugs, still we are facing problems with the cancer patients. There is no concrete evidence or there is no concrete treatment options for the cancer patients, especially the advanced stage. So why? Because still there are gaps. Still we need to go for a next level, to go for further studies, how to address the current problems, to treat the patients. So translating research is one of the is a research approach, is a really is a have a high impact compared to other conventional research studies. So including of patient care, public health practice, clinical approach, patient safety, all this translation research, which is very important to address all gaps and unmet medical needs. Translational research also is a dynamic and essential process and various stages from the basics to the clinical implementations, ultimately contributing to the improved health outcomes and to explore more medical knowledge. So these all importance of translation research. These are my references. I never focus on particular disease conditions. I wanted to insist why the translational research is important. Whatever we do, lab research, how we going to translate. So we should have the future directions, the future perspectives. then only we consider as a meaningful research. With this collaboration, which is very important, I do collaboration with many scientists in overseas, also in India. These are all my academic and research collaborations so far I have done with many eminent researchers. Before I was working Monash University, Malaysia. Also, I have collaborated with Monash University, Australia, and here just now before me, it's Kamal Dua, is Dr. Cam. He is one of my collaborators, and Professor Rajiv Dia. So many I have collaborators. Also, I do collaborate with China. Also, I am one of the adjunct professor in China University with the pharmaceutical science. Also, I do some effective MOA. It's not like just on a paper. We really have some good collaboration with the Indian universities, including Annamalai University, where I studied. Also, we do collaborate with many other institutes. So I have connected with Monash University, Australia. We have more than 4 projects ongoing. Also, I connected with IIT Bombay, Indian Institute of Technology. We have collaborative research projects. This is some kind of charity work. I do charity sometimes. When the disaster comes, 2019, we do charity works during visit China. It's a All India Institute of Medical Science during 2024 this year. It's a month of August. I visited AIMS University in the Department of Pharmacology. So we do have collaborations with the pharmacology departments. Also, this is my university, which is Kuala Lumpur. It's a Sunway University. These all my students. Lab is a center. He is my dean. He's a dean of medical and health sciences. Left side is a research dean with me. So likewise, during National University of Singapore, I visited Singapore University. It's a NUS. I have presented my research work. He is Gavin is a director of pharmacology. I do collaborate with Gavin. I am one of the advisor for their Master of Pharmacology program. So there are many opportunities to have a good collaborations. He's my senior, is a Professor Murali. He's working Auburn University, USA. Also, we have a good collaborations with Auburn University. Center is Prof. Cook. He's a vice president of alumni and engagements. And also Dean is a Professor Shah Jahan. He is from Monash University. So these are all my students during PhD final presentations. These are lab activities, lab workshops. Always we do lab workshops with my students and my collaborators. Is my students. Is during Monash convocation. These are all my students. This is during IIT visit is a Prof. Ashutosh. He's a bioscience professor. We have one research projects, ongoing research projects. Through that, we recruited one PhD student, still ongoing PhD studies. Likewise, we do have many collaborations. In 2019, I received a fellow of Malaysian Scientific Association from the society. It's a more than 70 years they have the society. Also, I have given research insights and research opportunities how to collaborate with IIT Bombay, Indian Institute of Technology, Powai, to have how to collaborate in the how to match align with those who are working on animal modeling. So during the research meet, we had good discussions with each other. From Australia, there are many researchers are attended these meetings. So these all my lab work, weekly, every week. Still, I'm going to animal lab. Minimum, I spend minimum quite hours, like 8 hours, 10 hours. I can spend still. So these all my one of the potent research team, like collaborators. Professor Kamal Dua is from Australia. Just now he presented before me. With his team, they visited Monash University when I was working before. to have is a possible research collaborations during visit. So these all my China visit to have workshop and hands-on trainings and workshops. So this is my university, Sunway University is a president. President is Professor Popima. Before he was president of Groningen University, Netherlands. Now he's working as he shifted to Sunway. Currently, he is the president of Sunway University. So with my team, thank you everyone for listening my short presentation. Thank you everyone for giving this opportunity. Really, it's a honor and privilege to have with you all and give my short presentation. Thank you.
Thank you, Professor Guerte, for your nice presentation. Over to you, judges, for any questions.
Good morning, sir. I am
Good morning.
Amrutwalli from the Institute of Pharmaceutical Sciences. Sir, it's really a nice and elaborative session to hear from you this morning with your very much detailed presentation on translation, research, bridging, science and practices. Sir, here is my question. Nowadays, we are hearing so much about artificial intelligence. Is there any chances utilizing this in pharmacy or hospitals for individual patient health data storage for further easy access? If we can use artificial intelligence, is there any data privacy problem?
Good question. It's a Miss Shaik. Who is asking question? Who is the name of?
Amrutwalli.
Amrutavalli is the good question. Artificial intelligence is a very important. Nowadays, we can use only for, there are some restrictions. We can use for predictive analysis, but we cannot go for real findings. Maybe we need a lot of evidences and artificial intelligence we can go for only for predictive analysis, predictions. Based on that, we can get some ideas. With that, we can go for some kind of evidence based or clinical research. But in the artificial intelligence based itself, we cannot take 100% as a research outcomes for clinical practice. Impossible. We do have some clinical evidence. With artificial intelligence, we can take a predictive analysis. Let's say example. Okay, if I'm doing animal modeling, maybe based on animal studies, maybe I can take animal movements. Maybe I can take blood parameters or biochemical estimations. From the data, I can get some predictive analysis. Maybe from the data, I can get some research conclusion. So this is maybe example, I can take only 30 animals. Maybe with the 30 animals, I can conclude with the 30 animals, I can conclude the results of the study. Okay, the final result. But AI is very helpful. We can expand the number of animals. Example, with the predictions, maybe we can conclude with the 30,000 of animal also without using animals. With the basic evidence, example, 30 animals results I have. Maybe with the artificial intelligence, maybe we can predict 30,000 animal results. Are you understand?
Yes, sir.
So these kind of predictions we can go for, but based on the artificial intelligence, we cannot move on to the clinical practice. Impossible. We must go with the clinical studies.
Thank you for your detailed explanation.
Thank you, Amrutavalli. Thank you.
Good morning, sir. This is SKF in Fatima. Be from Vijay Institute. Sir, it's really fortunate enough to hear from you digital research, bridging science and practice. Sir, here is my question. Recently, we heard about personalized medication or tele. Is it really possible to completely study individual subject and provide personalized medication without any contradiction?
we can be done, because based on the research, precision medicines is possible. But we have to categorize. It depends on the type of cancer, or depends on the type of disease modeling. So when we go for the specific modeling, so we need to include the precision approach. So the precision approach, why we need to go for precision approach? Some group of people may not get proper therapeutic responses, maybe genetic variations. So with the precision models, we can easily identify the genetic variations. So when we address these kind of variations, the patients, try to avoid inappropriate medications. Also, they used to get proper medications to get the recovery effects. So precision can be possible. We can go with a large population also.
Okay, sir. Thank you for your explanation.
Thank you.
Good morning, sir.
Good morning.
Sir, myself Krupa Mayi, Assistant Professor, Vijay Institute of Pharmaceutical Sciences.
Okay,
Sir,
madam.
it's really fortunate enough to hear from you regarding translational research bridging science and practice. And my question here is regarding practice. As you're mentioning that we have so much of development and usage of translational research, and getting that into practice is really a challenging. We are hearing regarding personalized or tailoring medication. And we recently heard that how easily we are getting money from the ATMs, that easily we can access to the medicine. And in our scenario, we are hearing mostly in about suicidal cases or most of the misusing of medications. If we give that much access to the individuals, those who didn't have any idea regarding medicines or their contradictions, for example, if we take antibiotics, we are getting more of resistance because of lackage of knowledge on that. If we give this much of access of medicines to the patients or general public, will we face any sort of challenges? Development is different. Getting that into practice will also give us so much of challenge as we are into this pharmacy field.
All right. Actually, there are many challenges. Inappropriate medication dispensing. So regulatory board also have to look for this kind of issues, because other countries like even India, Malaysia and Singapore, without prescription we cannot buy any antibiotics. But India is still we can buy OTC, over the counter. Is sometimes they do not know the effect of antibiotics, antimicrobial resistance. If they take these kind of inappropriate medications, sometime to develop the antimicrobial resistance. This kind of resistance, it may getting worsen for the patient itself, may not accept other treatment effects. Sometimes it may go for sepsis or it's dangerous events, adverse events. So that whenever we have to strengthen, it means we need to follow we have to implement certain policies to avoid this kind of to overcome the antimicrobial resistance. How to overcome antimicrobial resistance? Inappropriate, we should prescribe the appropriate medications based on the lab report or based on the diagnosis. Based on the symptoms, we should not prescribe any antibiotics. It's very dangerous. Based on the symptoms, based on the lab report only, we must go for antibiotics prescription. Also, the doctors also have to take a serious. It means take more concern to avoid antibiotic prescriptions for symptomatic responses or symptomatic responses from the patients. It means that if any patients come to the clinics, they should have proper evidence what kind of infections with the patients. Based on that only they go for antibiotic prescription. Otherwise, definitely it may chance to develop the antibiotic resistance. Also, we must encourage for combination therapies. Certain infections we cannot treat with only one antibiotics, because the microorganism is very strong. So we must go with the appropriate or combination treatment for the patients. Sometimes the doctors also do not know, or may not, the doctors may not identify properly, or they never do proper diagnosis. Based on the symptoms, if they prescribe any medications, also will develop the resistance. So antibiotic resistance is a high threat in India. Not only in India, the global threat. But India also is very, very dangerous because of high populations. There is no proper regulation. Sometimes OTC itself, we can get some antibiotics. These all sometimes cumulatively or collectively, it may chance to develop a lot of side effects. Sometimes, they may not get the proper treatment recovery also in the any serious conditions. So these all difficulties are there, practical difficulties. But antibiotic resistance we should avoid. It means how to avoid. One is they should not encourage to take self medications unless without emergency, or maybe they can take only NSAIDs or certain allowable drugs only. Common drugs they can take, but they should not take without a doctor advice or without a doctor prescription. They should not take any antibiotics themselves. Self medication sometimes is very dangerous for common people. So these are the measuring points. Maybe we try to avoid the antibiotic resistance.
Yes, sir. So here my question again is, if we are giving personalized medicine, is there any problem regarding that? Without any knowledge on the medicines, will they take this easy access for medication?
Regulatory guidelines, we can go for any research, but without regulatory guidelines or without proper procedures, we should not go any research, including precision medicine.
Yes. Yes.
May I answer your question, madam?
Sir, actually, I'm not asking detailed on antibiotic resistance, but I want, if we are developing these much of emerging research on personalized medication, tailoring medication, 3D printing techniques, all these techniques, will they cause any sort of effect, negative effect on pharmacy or on the market regarding patients, those who didn't know anything about medication? Will they misuse these medications by these type of techniques?
These all approaches, main reason is to avoid the resistance, to avoid the inappropriate side effects. It means that to avoid the side effects, to avoid the any adverse events for the patients, so that we need to go for these kind of approaches. These all approaches are good for clinical studies, are good for the patients. It's not there is no negative impacts, so that we advanced level we are going for approaches, different approaches, to try to avoid the toxicity, to try to avoid the resistance, to try to avoid the unnecessary complications.
Okay, sir.
Okay, thank you.
Thank you, Dr. Vetri,
Thank you, Prof. Rajiv
for
Gya.
a nice discussion and to answer the query. Now we will move to our next speaker, which is none other than Professor Parang from Chapman University, Irvine, California. Welcome, Professor Parang. I invite the co-convener to read a few points about Professor Parang, and then we will continue with the lecture. Thank you.
Okay, sir. Now I would like to invite Mr. Sri Krupamai, Assistant Professor, Department of Pharmaceutical Chemistry, to share the profile of our keynote speaker, Kikavas Parang.
Good morning, everyone. I, G. Krupamai, Assistant Professor, Department of Pharmaceutical Chemistry, Vijaya Institute of Pharmaceutical Sciences for Women. I feel honored and privileged to present the profile of today's keynote speaker, Dr. Kikavas Parang, PharmD, PhD. Currently, Dr. Kikava Parang is a full professor in the Department of Medicinal Chemistry and Pharmacology at Chapman University, School of Pharmacy, and Smith College of Science and Technology in Irvine, California. Coming to educational background of Dr. Parang includes, Sir earned his PharmD degree from Tehran University of Medical Sciences in the year 1989 and completed his doctoral degree in medicinal chemistry at the University of Alberta, Canada in the year 1997. Followed by, Dr. Parang also conducted his postdoctoral research in the Department of Chemistry at the University of Alberta. Further, Sir also pursued his postdoctoral studies in bioorganic chemistry and pharmacology at Rockefeller University and Johns Hopkins University. Dr. Perang's working experience includes, in October of 2000, Sir joined the College of Pharmacy at the University of Rhode Island, where Sir became an associate professor in April of 2006 and a full professor in July of 2008. During his tenure, Sir served as the program coordinator for the Rhode Island IDEA Network of Biomedical Research Excellence (NIH) program, which had a budget of $20 millions. In Indian currency, it's about 2 crores budget during 2012 to 2013. Dr. Perang joined Chapman University as the second founding member, assisting in establishing the first pharmacy school in Orange County, California. Sir served as Associate Dean of Research, Graduate Studies and Global Affairs during the period June 2013 to June 2020, and also served as Associate Dean of Research, Innovation and Global Affairs during June 2020, March 2022 at the School of Pharmacy. He is also an affiliated faculty at the Division of Nephrology and Hypertension, School of Medicine at the University of California, Irvine. Sir was also a visiting scientist in the Department of Pharmacology and Molecular Sciences, School of Medicine, Johns Hopkins University during 2002-2017. Dr. Perang has supervised 28 postdoctoral fellows and visiting scientists, additionally mentored 31 graduates, including 14 PhD, 17 MS, as well as 61 UG candidates. Dr. Perang serves as an editorial board member or editorial advisory board of several journals, such as the Journal of Biological Chemistry from 2025 to 2029, Molecules and Frontiers in Nanotechnology Journal as well. Dr. Perang's research mainly focuses on the intersection of chemistry and biology, with particular emphasis on medicinal chemistry, organic chemistry, nanomedicine, and drug delivery. Sir's work involves applying synthetic organic chemistry to biological challenges. Dr. Perang's current research areas include utilizing cyclic and hybrid cyclic linear peptides for drug delivery, designing peptides with antibacterial and antifungal properties, developing multifunctional agents with antiviral, anti-cancer, and antibacterial activities, and also creating inhibitors for protein tyrosine kinases. Dr. Perang's contributions have resulted in 245 peer-reviewed publications, 23 issued or pending patents, 201 meeting abstracts, and 63 invited oral presentations. With an illustrious career marked by groundbreaking research and an unwavering commitment to advancing the chemistry and pharmacology fields, Dr. Perang stands as a leading authority in various pharmaceutical emerging scenarios. We are very much honored to have Dr. Perang spending his valuable time with us today and looking forward to thought-provoking insights that promise to inspire and enlighten us all with his talk. Thank you for giving me this opportunity. With this, on behalf of APP and Vijay Institute of Pharmaceutical Sciences for Women, I invite our keynote speaker, Dr. Kekavos Perang, to deliver his talk. Thank you, sir. You're welcome.
Thank you so much for your good introduction. Everyone can hear me?
Yes, sir.
Okay. So I have shared my presentation. First of all, I would like to thank Association of Pharmacy Professionals for invitation and also the president of the association. And also I would like to thank Dr. Dahia for the warm invitation. So let me tell you a little bit about my research. The title of my talk is about development of cyclic peptide doxorubicin conjugate to overcome multi-drug resistance and reduce toxicity. So Chapman University is at Southern California, and I would be delighted any of you who visited California to visit Chapman University and give a talk also. So let me go to my introduction and problem that we have. As you know, there is a major problem for cancer, and we usually have about 10 million deaths per annually, and mortality is very high. We have about 20 million cases. So if we consider COVID-19 during the last 4 years of the epidemic, we have about 6 million deaths. So we're talking about 10 million deaths every year. And in patient, female breast cancer and lung cancer are the major cancer, and Dr. Dua talked about lung cancer, so that's a major problem. The major problem also we have in cancer chemotherapy, we don't have a specificity for drugs, and we get a lot of toxicity, and many cancer drugs also develop resistance, and they have undesirable pharmacokinetic property. So if we look at the IV injection of cancer drug until they get to the target, so there are major problem for distribution of the drug. Major situation we have going to the wrong target, and also the penetration through the endothelium, and eventually getting to the tumor cell. We may have cell wall permeability and degradation of the drug, and efflux of the drug from the cancer cell, and eventually resistance. So these are the major problems for many of the cancer cells. So I just highlighted the cytotoxicity, endothelium permeability, cell wall permeability, and eventually many of the cancer drugs are acting in the nucleus of the cells that interacting with the DNA. So they have to pass through the nuclear wall. So as you heard today from Dr. Dua, there are many different approaches for cancer drug delivery, and you heard about liposome and nanoparticle. And there are other development peptide-based drug delivery. We have also using the albumin-based and red blood cell erythrocyte drug delivery, antibody drug conjugate, peptide drug conjugate. So all of these approaches are emerging approaches that have been used for several drugs, and each one of them have major advantage and disadvantages. So just to highlight some of these, when we're talking about liposome are phospholipid bilayer that can be used for delivery of hydrophobic drug and hydrophilic drug, and we can encapsulate both type of drug inside the liposome. The problem with the liposome is that they may have a very short half-life, and they may have not a very long stability, and we have a leakage of the drug outside the liposome. And the immune system also has a reaction with some of these liposomes. There is another approach using polylactic glycolic acid. This is a copolymer of lactic acid and glycolic acid. The problem with the PLGA is also degradation of the PLGA to glycolic acid and some cytotoxicity. Another recent development is solid lipid nanoparticle and lipid nanoparticle that are used for cancer cells. In solid lipid nanoparticle, we have a solid lipid and a lipid bilayer that usually is a monolayer. So we can use this for encapsulation of, we can use that to increase the stability. But unfortunately, the loading capacity and encapsulation capacity of solid lipid nanoparticle are also limited. And as you know, lipid nanoparticles have been used for also antiviral vaccine, that we had a COVID vaccine recently, mRNA vaccine. Another approach is the cell-penetrating peptide that I'm going to talk about today. So cell-penetrating peptide, as you know, peptides are a compound that composed of different amino acids, and they can be divided in different approaches based on the physical-chemical property. We can have cationic, hydrophobic, and a combination of both cationic and hydrophobic amino acid. We call it amphipathic. We may also have linear peptides, so we have a multiple monomer of building block of amino acid, or we may have a cyclic peptide, means that the head and tail of the amino acid peptide chain are connected with each other. Or we may have a hybrid peptide, which means that we have a cyclic peptide and linear peptide attached to each other. Or some of these cell-penetrating peptide, they can be naturally protein-derived, some of them may be synthetic, or some of them chimeric, or a combination of these. So cell-penetrating peptide can be conjugated with the different cargo molecule like nucleic acid, protein, a small molecule, anti-cancer drug, and even imaging agent. So they can be used for delivery of many of different cargo molecule to different cells. And my focus today is to use this cell-penetrating peptide to deliver anti-cancer agent. So generally, linear cell-penetrating peptide, they are short amphipathic peptide that they have a very high internalization into the cells. But unfortunately, they have a very short stability. Peptides have a tendency to be degraded because we have a lot of proteases in our body that similarly that they digest our food and protein, they can also digest the peptide. So, and on the other hand, they are easily synthesized and they are natural, but they are not very target specific. They can be degraded, sometimes they can be toxic, and they have a very short half-life. And some of the example of the linear cell penetrating peptide, as you can see here, TAT was one of the first cell penetrating peptide that was derived from the HIV, because HIV has a protein called TAT that has a very strong cell penetrating. And as you can see, this peptide has multiple amino acids. Each one of these letters shows one amino acid. For example, R shows arginine. And many of these cell-penetrating peptides have positively charged amino acid like arginine. PEP1 is another one that has WW is a tryptophan, and they are kind of amphipathic. So you have arginine and lysine that are positive charge, and tryptophan that is hydrophobic. So we have amphipathic. So and many other cell-penetrating peptides that are linear. So my approaches has been to develop cyclic cell-penetrating peptide. When you cyclize the linear cell-penetrating peptide, you make them much more stable to proteases in our body. On the other hand, you also generate more cell permeability compared to linear peptide. And many of these linear cell-penetrating peptide, when they get into the cells, they have to go inside an endosome, inside the cell, and they have to get out of the endosome and release the drug. And sometimes they get trapped inside the endosome, inside the cell, and they never release the drug. So many of cell-penetrating peptides bypass the endosome, or they can easily escape the endosome upon uptake. So in my research, we have developed two different types of cyclic peptides. First, we have an alternate hydrophobic and cationic positively charged amino acids that are tryptophan and arginine. So these are alternate. So we call it WR. Peptide means W means tryptophan, R means arginine. If we put subsequent positively charged amino acid and subsequent hydrophobic amino acid like tryptophan, we call it W4R4. And interestingly, when we have alternate amino acid of positive charge and hydrophobic, we get a cell-penetrating effect. But when we put all the hydrophobic amino acid in one side and all the positive charge amino acid on the other side, we get antimicrobial peptide. So we were able to develop two different types of compound using cyclic peptide, cell-penetrating and antimicrobial peptide. Okay, so my focus today will be mostly on the cell delivery, not antibiotic. That's another topic and interest that I'm working on. So for this type of WR peptide that are alternate positive charge and hydrophobic, we have been able to use them for different applications. And my focus will be today on the doxorubicin prodrug and conjugate. So we have been able to conjugate this peptide with the doxorubicin. Doxorubicin is the anti-cancer drug and is a gold standard in cancer chemotherapy. Unfortunately, doxorubicin has a lot of toxicity, and major toxicity is a cardiotoxicity. So the oncologists hesitate to use doxorubicin for people who have cardiovascular diseases. And also another major problem with doxorubicin is develop very rapid resistance in cancer cells. So, and after a while, the patient is not responding to doxorubicin. So cardiotoxicity and resistance is a major problem with the doxorubicin. So we wanted to use this kind of cyclic peptide and also hybrid peptide, cyclic linear peptide, to develop doxorubicin conjugate and be able to bypass the toxicity and also reduce the resistance of doxorubicin. But this peptide has been used for many other applications. We have been using it for protein kinase inhibition. We also attach it to other anti-cancer drugs, including curcumin. We have been using for delivery of nucleic acid like siRNA, CRISPR, and phosphopeptide. We may able to generate gold nanoparticle. We have been using to stabilize the protein and also target the nucleus of the cancer cell. So how this works? So even I'm talking about tryptophan and arginine type of peptide that are cyclized. These are a small peptide, and they get self-assembled with each other. And as you can see in this electron microscopy picture, a small peptide generates nanoparticle, and these nanoparticles are self-assembled with each other and generate even larger particle. And this larger particle can be used for the encapsulation of the drug. So we have a hydrophobic tryptophan group and hydrophilic polar residue like arginine. And these arginine residue are exposed to the water, and then hydrophobic group love to self-assemble with each other. And then we generate a nanoparticle, and we can encapsulate the hydrophobic anti-cancer drug inside the nanoparticle and deliver it to the cancer cell. So it's like a Trojan horse. So we can secretly deliver the anti-cancer drug into the cancer cell. So to see whether this is working, we develop an antiviral drug first. This is a lamivudine anti-HIV drug. And we use a linker and attach it to the fluorescent. And we wanted to see whether we can use this cargo molecule to different cells. So we use different type of cyclic peptide. And as I told you, we use hydrophobic and positive charge amino acid. But then we also use other type of peptide that like phenylalanine, we also use alanine, we use even negatively charged cyclic peptide. We wanted to see whether our hypothesis is correct and the presence of hydrophobic and positive charge change the uptake. So because this cargo molecule has a fluorescent, we can measure the uptake. So in the presence of the WR4 and WR5, that is the 4-tryptophan and 4-arginine, we see increased uptake of the fluorescence labeled lamivudine. But in other cyclic peptide, we don't really see much improved uptake. So that shows that our hypothesis that the combination of this cyclic peptide with the antiviral drug can be used for delivery. And we can also use the fluorescence microscopy. If you use on the fluorescence label the drug, it doesn't get into the leukemia cell very efficiently. But when in presence of WR4 and WR5, you can see the high fluorescence of the cancer cells. So we can actually deliver the drug into the cells and track it using the fluorescence. And if you look at the even fluorescence labeled phosphopeptide, alone phosphopeptides are not able to get into the cell because they have negative charge, and the cell membrane have also negative charge. So we have a very low uptake. But when we have the cyclic peptide, we see very high fluorescence of the peptide inside the cell. And DAPI is a stain that's used for the staining the nucleus of the cells. So when we use DAPI, we can see where are the nucleus. Okay, and Texas red is another stain. We can use the cytoskeleton and the membrane of the cells. And then when we combine all these stain, you can see the fluorescence labels is localized inside the nucleus. So blue and green are overlapping and coalesce inside the nucleus of the cancer cell. So if I show you only the fluorescence label phosphopeptide, as you can see, the uptake, there is not much uptake inside the cell. But when I added the cyclic peptide, and you can see that the fluorescence label green is actually inside the nucleus of the cell, and we can actually see this with the three-dimensional image. Okay, so how about the cytotoxicity of this peptide? So when we look at the cytotoxicity of this peptide alone in different cell lines like leukemia, colon cancer, breast cancer, ovarian cancer, we don't see any change in cell viability up to 100 micromolar. And where this peptide goes when we use the peptide alone. So we decided to conjugate this peptide with a fluorescence label and see where it goes. And we incubated different cell lines like ovarian, colon, and breast cancer. And as I told you earlier, the DAPI is used for staining the nucleus. So this is only the peptide, the fluorescence label peptide, so it's a green fluorescence. And this is the nucleus, and when we combine this, we can see wherever there is a peptide, also there is nucleus. So it means that this peptide actually is a nuclear targeting. It goes to the nucleus of the cancer cell, and we can conjugate it with anti-cancer drug like doxorubicin to deliver it to the nucleus of the cell. So we can bypass the endocytosis and endosome. That is a major problem. So initial conclusion that I have that the peptide that containing tryptophan and arginine, they can self-assemble, generate nanoparticle, and then they can be used for as a molecular transporter, and then we can use this for delivery of different type of a small molecule or negatively charged molecule. So now I'm getting to the major part of my talk, the delivery of doxorubicin. So doxorubicin, that you as a pharmacy professional, you know, doxorubicin has a red color. And you can see the drug is used as an injectable form in hospital setting. And it's an anthracycline drug. It's intercalated inside the DNA of the cancer cell. But also it has another mechanism. It can inhibit the enzyme called topoisomerase 2. Topoisomerase is the enzyme that is used to relax the super cold DNA, because when the DNA is twisted, sometimes it's not able to relax and separate, and we don't have transcription and translation. So topoisomerase cut down the DNA and make a nick DNA, relax it, and then seal it again. So we have a nicking and resealing by topoisomerase. So the doxorubicin inhibit topoisomerase and for both double-stranded DNA and reduce the relaxing of the DNA in the cancer cell. So they cannot be relaxed and they cannot replicate. But the problem with doxorubicin, as I told you, is the cardiotoxicity and doxorubicin resistance by cancer cells. And this is showing the topoisomerase that is cutting the double-stranded DNA in the chain, and then doxorubicin intercalated this, and also doxorubicin interact with topoisomerase 2 and inhibit topoisomerase. In terms of doxorubicin resistance, the major problem is that there is a drug efflux pump that gets rid of doxorubicin after it gets to the cancer cell. So one of the major pump is a permeability glycoprotein and other type of multi-drug resistance that are getting rid of the doxorubicin. There are other mechanisms of resistance like a decreased uptake and change in apoptotic pathway, but the major problem is the drug reflux. But if you look at the doxorubicin mechanism, Usually doxorubicin integrated into DNA, and then, as I told you, inhibit the topoisomerase, and then we have reactive oxygen species that generate activity in the cancer cells also. But on the other hand, doxorubicin generate reactive oxygen species like superoxide oxygen and halogen peroxide, that these are actually interacting with the heart cell and causing a lot of toxicity. So if we look at the major site of interaction of doxorubicin, the DNA integrating domain, and also there is the topoisomerase interacting domain. So we cannot really touch this part of the molecule because these are important for interacting with DNA and also interacting with the topoisomerase. Only functional group that we were able to use is the primary hydroxy group for conjugation with our peptide. So we made different type of conjugate of doxorubicin using cyclic peptide, a small cyclic peptide, larger cyclic peptide, hybrid cyclic peptide means you have positive charge cyclic and hydrophobic chain and attach it to doxorubicin. We also use different type of linker. We use a disulfide or amide or ester. So this is another hybrid cyclic peptide that we use for conjugation with doxorubicin. So I'm going to give you some story about some of these. So many of these has been published in different journals, and as you can see, they have antiproliferative activity and high uptake and retention. Some of them actually is improving the activity against resistant cell and reduce the cytotoxicity against heart cells. So, and they bypass the endocytosis. So let me start with a small example that we have a cyclic peptide that has a phobic and positive charge, the WR peptide. We have a linker and we attach it to the primary hydroxy group of doxorubicin. So when we use doxorubicin alone in the cancer cell, this is ovarian cancer cell, it has a very good uptake. After 1 hour, you can see doxorubicin inside the nucleus. You can see the red color of the nucleus. If we use the conjugate also, we can see high uptake of the conjugate and doxorubicin inside the nucleus. After 24 hours, as I said, doxorubicin has a very high efflux. So most of the doxorubicin is efflux out of the cancer cell. But when you have the conjugate, we still have a lot of retention and uptake of the doxorubicin. So it means that the conjugate helping the retention of doxorubicin inside the cancer cell. But it's not enough. We need to have also selectivity. So how we can target doxorubicin into the cancer cell. So many of the cancer cells have different type of protein like a folic acid receptor that is overexpressed. So we can attach a folic acid to our peptide to target the folic acid receptor in cancer cells. Also, we want to have an improved stability of our particle. So we usually use polyethylene glycol. Polyethylene glycol is usually used for capping of the protein, but also we can use the to cap our molecule. So it has the longest circulation in blood and improve stability. But we want to also see where our molecule goes. So we use the fluorescent tag to our peptide to see where it goes. So we design a peptide with a cyclic peptide. It has a folic acid to target the cancer cell. The peptide as itself has a cell penetrating effect. We attach a polyethylene glycol 5000 to escape the liver excretion and enhance the stability. And we had a fluorescent tag CY55 to improve the imaging. And if you look at the mice study, we have two types of tumor. On the right-hand side, we have a tumor that overexpresses folic acid. On the left-hand side, we have a tumor that doesn't express the folic acid. So we have the HEK293T and HeLa tumor in both xenograft mice model. And when we use the conjugate, as you can see, most of the conjugate is localized in the right tumor. we don't see much uptake in the left tumor because the left tumor doesn't have the folic acid receptor. And if you separate all different organs, you can see that most of the compound is inside the Hilo tumor and not in the HEK tumor. That doesn't have a high folic acid. You still see some uptake in the liver, lung, and kidney. That is usually what we see for many drugs for the excretion of the compound, but we have a high localization in the tumor cell. So this conjugate we had didn't have any doxorubicin. The next step was to conjugate the doxorubicin to our peptide. And also we have an imaging agent, polyethylene glycol, and targeting moiety that is folic acid. So we use this imaging targeting drug and we inject it to the mice. And within 1 hour, we were able to localize most of the compound inside the tumor. But then it started eliminating in the bladder and getting eliminated from the body. So you can see that there was a high localization of the compound in the tumor after 1 hour. And if you look at the 3 hours, sorry about that, look like. I have to share it again in just a moment. Sorry for interruption. Let me go back to my slide again. So after 3 hours, you see most of the compound goes to the bladder. So we can actually measure this. The tumor uptake goes low, and then bladder excretion goes higher. So how we can improve the more uptake in the tumor? So we use different type of peptide. We increase the number of amino acid, and we saw that if we increase the number of hydrophobic and positive charge amino acid, we have much higher uptake of different cargo molecules. Compared to 5 amino acids, we have 9 arginine and 9 tryptophan. We have much higher uptake. So we use WR9 and conjugate our peptide with the doxorubicin. And as you can see that if we look at different cancer cells, if you look at doxorubicin alone, this is MDA-MB231 triple negative breast cancer cell. This is the viability of the cancer cell. In the presence of conjugate 1 micromolar, 5 micromolar, and 10 micromolar, we have much better antiproliferative activity compared to doxorubicin. Similarly, here in the MCF7, we have much higher cell antiproliferative activity and less cell viability here. After 72 hours, we see that the antiproliferative activity is much higher than 24 hours. It means that the conjugate is slowly hydrolyzed to doxorubicin at a longer period of time. And we can also see this using fluorescence microscopes. So if you look at the MDA-MB231, the uptake of the doxorubicin, and then the physical mixture, we also have uptake. But when we have the conjugate, we have significantly higher uptake. How about doxorubicin resistant cell? So when you look at doxorubicin resistant cell, So doxorubicin is not active against resistant cells, so we only see limited antiproliferative activity. But when we have a conjugate, we have significantly antiproliferative activity in doxorubicin resistant cells. So means that this conjugate is blocking the efflux of doxorubicin inside the doxorubicin resistant cell. And you can see this in the fluorescence microscope. Doxorubicin is not uptaken by the resistant cell. If you have a physical mixture or conjugate, you see a lot of doxorubicin inside the nucleus of the cancer cell. So as I mentioned, also endocytosis is a major mechanism that blocks the uptake of the many cancer cells. They may go to the endocytosis, clathrin or keratinoid mediated or micropinocytes, and then they get trapped there and they're not released into the nucleus. So if we use a different inhibitor of endocytosis inhibitor like chlorpromazine, chloroquine, nystatin, methyl beta-cyclodactyrin, we can see whether the doxorubicin is still uptaken in the presence of inhibitor. So if we use a conjugate and then we use the inhibitor with the conjugate, we see the uptake is not affected by the inhibitor. So means the endocytosis inhibitor are not blocking the uptake of the doxorubicin conjugate. And we can also see this in the fluorescence microscope with chloroquine, chlorpromazine, cyclodextrin, we still have high uptake of the conjugate into the cancer cell. And how about the stability of the compound? So when we look at the doxorubicin conjugate, we have a ester bond and amide bond. These are going to get breakdown and then doxorubicin release. So if we use the human plasma, we can see the uptake and then a slow release of doxorubicin and from the conjugate and a slow degradation of the conjugate. And we can see this with the HPLC, doxorubicin conjugate reduce and the free doxorubicin increase after 12 hours, 24 hours, and eventually after 48 hours, we have only free doxorubicin, very limited doxorubicin conjugate. Another part of my presentation I promised was to talk about cyclic peptide, linear peptide conjugate, the hybrid peptide. So this hybrid peptide, cyclic linear peptide, were conjugated with doxorubicin, and we look at the uptake in the cancer cell and heart cell. We were able to see that the uptake in the cancer cell is very high, but in the heart cell is very limited. And we can really see that with using the antiproliferative assay in leukemia cell, stomach cancer cell. So as you can see, doxorubicin is very effective. And our conjugate is also very effective against similar cancer cells, especially after 72 hours. So if we look at the doxorubicin, it has a very high uptake in these cancer cells. But then when we have the conjugate, we also even have much more higher uptake of doxorubicin in the cancer cell. So we are able to replicate and even improve the anti-cancer activity of doxorubicin. But when you look at the heart cell doxorubicin has a very high toxicity. As you can see, you see 60% less viability. But then our conjugate is not very toxic in the heart cell. Similarly, in kidney cell, kidney doxorubicin is very toxic. The cell viability is significantly reduced, but our conjugate is not toxic. Okay. And if you look at the fluorescence microscope, you can see that the doxorubicin get into the heart cell significantly, but our conjugate is not able to get to the heart cell. That shows that that's why we don't have toxicity. And we can actually see also the activity of doxorubicin in resistant cell and compare it with our conjugate. So pre-doxorubicin and conjugate have very similar IC50. They can both very effective. But when you use the MDA231 that is resistant, pre-doxorubicin is not very active, but our conjugate is almost 5-fold more potent. Also, another type of resistance that pre-doxorubicin is not very active, but our conjugate is 5-fold more potent. So it shows that the compound is helping the doxorubicin to bypass the resistance mechanism. And so let me continue my talk. So if you look at using the endocytosis inhibitor, you can see that in the presence of endocytosis inhibitor, you can still see the high uptake of the conjugate inside the cancer cells. And we can quantify this. We don't see any blocking of endocytosis in the presence of the endocytosis inhibitor. So the cellular uptake is not affected by this. And finally, we also developed a hybrid cyclic peptide that have also antimicrobial property. So if you have an antimicrobial peptide on the cyclic part, an antimicrobial peptide on the linear part, and we conjugate with each other, surprisingly, when we tested the antimicrobial peptide of this hybrid peptide, it didn't show any antimicrobial peptide property. But when we attached these two antimicrobial peptides, we saw significant cellular delivery. So it was able to develop a cyclic cell-penetrating peptide. So if you look at here, doxorubicin has a good uptake, very limited uptake in doxorubicin resistant cells. But then when we have the conjugate and the doxorubicin physically mixed, we get very high uptake in doxorubicin resistant cells. So we were able to improve the uptake of doxorubicin within the physical mixture using two antimicrobial peptide, hybrid peptide to the cancer cell. And if we attach a fluorescence label to our hybrid peptide, and we wanted to see whether it has a time-dependent or concentration-dependent uptake. As you can see, the fluorescence labeled peptide improve uptake as we increase the concentration, and also time-dependent, 5 minutes, 30 minutes, and 60 minutes. So it gets to the cell very immediately. Within 5 minutes, we get very high uptake of the peptide inside the cell. And as we increase the concentration, also the uptake is higher. So now we use this hybrid peptide that included two antimicrobial peptides attached to the doxorubicin. And first we tested the triple-negative breast cancer cells and breast cancer cells. As you can see, doxorubicin effective by itself. But when we have a conjugate, we get much higher efficiency and antiproliferative activity in different type of breast cancer cells. So the conjugate is able to deliver the doxorubicin into the breast cancer much more efficiently. On the other hand, doxorubicin is not effective against resistant cells, is not generating any antiproliferative activity. But when we have the conjugate, we have significant antiproliferative activity. So the conjugate was still active against the resistant cell. And interestingly, when we look at the doxorubicin against heart cell, as you can see, the heart cell are very sensitive to doxorubicin. So we get very cardiotoxicity because of this. But then our conjugate was not toxic against heart cell. So we didn't see any significant cytotoxicity up to 8 micromolar against heart cell. So what's the mechanism? So the heart cell have a receptor, we call it the ryanodine receptor. And ryanodine is, when it's bound to the doxorubicin, it generates a calcium leakage. And calcium leakage causes the calcium hemostasis and death of heart cell. In case of our conjugate, it has much higher binding affinity to this ryanodine receptor, but it's acting as the antagonist. So it binds to the ryanodine receptor and block the calcium leakage from the ryanodine. So that is the mechanism that we believe that prevent the doxorubicin cardiotoxicity. And if we look at the peptide alone, it doesn't get to the cells. Doxorubicin is not very efficient getting into the cancer cell that are doxorubicin resistant. When we have the conjugate or physical mixture, we get very high uptake in the nucleus of the cancer cell. On the other hand, when you look at the heart cell, doxorubicin is getting high localization in the heart cell, but our conjugate is not getting into the heart cell. And if we can quantify this, in doxorubicin is not very effective against resistant cell, but our conjugate is much effective than doxorubicin and physical mixture. On the other hand, doxorubicin has very high uptake in the heart cell, but our conjugate has no efficiency against heart cell. And we can actually see what's happening using apoptosis pathway using flow cytometry. So we can see how the doxorubicin affecting the late apoptosis or early apoptosis after 24 hours and 48 hours. And also conjugate, when we have the conjugate, how it's affecting the uptake of the doxorubicin. So if I want to show this in a slide, in the resistant cell, as you can see, doxorubicin generating not much apoptosis in the resistant cells. But then when we have the conjugate, we have high early apoptosis and high late apoptosis and causing the programmed cell death in cancer cells. On the other hand, when we look at the heart cell, doxorubicin is generating a lot of early apoptosis and late apoptosis in the heart cell, but our conjugate is not generating any early apoptosis and late apoptosis in the heart cell. So that shows our compound is working in two problems that we had, heart cytotoxicity and also drug resistance. When we look at the serum stability of this compound, we see the half-life is about 14 hours, and we can measure it. The conjugate and pre-doxorubicin conjugate concentration goes down, and doxorubicin conjugate goes up. And we know that cancer cells have an acidic environment. So when we look at the acidic environment in the cancer cell, around 6.8, and you can see the compound in acidic environment, doxorubicin is released much faster compared to physiological pH. So that's another advantage. So we can consider that this peptide conjugate can release the doxorubicin in cancer cell much efficiently. And if we look at the intracellular hydrolysis of the conjugate, so when the peptide conjugate drug goes into the cell, and we look at the concentration of doxorubicin release, we see after 24 hours, the doxorubicin started release inside the cancer cell, and the conjugate reduced. Okay, so we can actually measure and do the cell lysis and measure the amount of doxorubicin inside the cancer cell. So let me end up my talk. So we made a lot of cyclic linear hybrid peptide that we can consider as a prodrug strategy. It can be effective against doxorubicin resistant cells. On the other hand, it can mitigate the cardiotoxicity. We can bypass the endocytosis, and then also we can have increased uptake of doxorubicin and antiproliferative activity and good half-life, and can be used for successful drug delivery system and overcome the permeability glycoprotein efflux mechanism. So let me thank some of my colleagues who work in this project. There are many people have worked during the last 2 decades. Many of them are Indian scientists, and some of the best scientists have worked in my lab, Dr. Tiwari, Dr. Mandal, Dr. Lohan. Some of them are already professor and department chair and director, or they work in the pharmaceutical industry. And also many graduate students and undergraduate students who have worked in my lab. And also a lot of funding agency, American Cancer Society, NIH, National Science Foundation, that during the last few years have supported my research. So by saying that, I would like to thank again the audience for listening to my talk. And currently I'm in Spain, visiting Spain, but I'm planning to visit India, Hyderabad in February, and hopefully able to network with scientists in India again. I have visited India many times, and I'm looking forward to my next visit. Thank you, everyone, and I'll be delighted to answer any question you may have.
Thank you, Professor Paran, for the wonderful talk. And I feel that this will be contributed toward the innovative pharmaceutical research for our young pharmacy professionals. So, uh, uh, I will ask the judges and participant for any questions for Professor Paran. Any question?
Good afternoon, sir.
Yeah. Hello, how
Sir,
are you?
hi, sir. It's really very much a pleasure to hear your talk. And myself, Krupa Mai, Department of Pharmaceutical Chemistry Assistant Professor, VIPW. Sir, it's really elaborative, and your research work is so much interesting. And here my question is, sir, you have emphasized on doxorubicin anthracycline antibiotic by combining with cyclic peptides to overcome multi-drug resistance and to reduce toxicity. So we have a few other drugs like donorubicin with this class. And have you got a result that by combining with these cyclic peptides, can we achieve completely this multi-drug resistance or to reduce toxicity? In general, we will hear that whatever the drugs we will take for cancer, they used to act on normal cells also rather than acting on cancerous cells. Without using these cyclic peptides, we are seeing those. You have combined the cyclic peptides with anthracyclines like doxorubicin. If we try to done with other moieties available, can we achieve that, sir?
Yeah, that's a great question. You mentioned donorubicin. We're working on donorubicin. We also work on epirubicin. These are all anti-cancer drugs that some of them are used in pediatric cancer, especially, for example, donorubicin is used for the small children.
Yes.
So we are working on that. For the epirubicin, we already finished the work, and we saw very similar result. The epirubicin also have cardiotoxicity, but when we have the conjugate, we reduce the toxicity, and also we bypass the resistance. So this is, and we have done this with the other anti-cancer drugs like camptothecin, paclitaxel, and curcumin. So in response to your question, this approach can be used for almost several anti-cancer drugs. We've done some animal study, but then again, how this goes in the human study, we have to do much more translational type of research, as you heard today. But based on the preclinical study we have, this is an effective approach for delivering of other cancer drugs also.
Yes, sir. Sir, we are using some computer-aided drug design and some in silico studies regarding our pharmacological or any of these studies. Can we utilize by combining this drug with the peptides, and can we done this on drug designing techniques using computer-aided drug design, or can we get these proteins or peptides from the protein drug bank, and can we create some docking techniques, or can we done any sort of research work on this?
Yeah, that's another good question. So usually docking and protein modeling are used to find out what type of ligand can bind to the binding pocket of enzyme or even in DNA. In this case, we already know where the doxorubicin bond. We know that it's bound to the topoisomerase. So in our goal is to generate a drug. Our goal is not to generate a new drug moiety because we already know the drug bound to topoisomerase, and when we conjugate it with the peptide, we are still getting the same binding pocket. So we are not really developing new drug. We're developing a drug that is on the market and improve the activity and reduce the toxicity and bypass the resistance. But in general, answering your question, if computer modeling is very effective approach when it's combined with the experimental approaches for docking. So if you have a anti-cancer target and you want to develop a compound, you can use visual screening of thousands of compounds inside the binding pocket of that compound. So if I had another anti-cancer drug that I don't know where it bound, I can dock it to the binding pocket and see whether you get some good binding energy, and then go and synthesize that compound and then see whether it really works. So this approach is used in the pharmaceutical industry and academia. But in our particular case, we use AI and computer modeling for other approaches, especially for antibiotic drug development. But for this particular project, we already know where it binds. So, and we already have a drug that is available. So we are trying to improve it. So we didn't use it for this approach, but that's a great question.
Yes, sir. Thank you so much for your detailed session on this, sir. Thank you very much.
You're welcome.
Thank you, Professor Parang, for your time, and we will definitely meet in future. So I will talk to you in separate session. So
Thank you
here
so
we
much.
close here we close our virtual 4 lecture scientific session, and now there will be break, and we will continue with our e-oration competition. And I will ask the convener and co-conveners to make me host of this Zoom so that I can lead the session at 1:15 p.m.
Sure,
Thank you all.
sir. Thank you, sir.
So you can make me host.
Yes, sir. Hello, Raju,
So
sir.
I'm the host now. I'm the host now.
No, sir.
So you can make me host so that I can run the session at 1:15 p.m.
Okay, sir. Okay, sir.
Sir Raju, sir, all the talks
Yes,
are
Kamal.
very informative.
Yes,
So very nice
thank you.
sessions you have arranged, sir. All the 4
Yeah.
sessions, even today I have completely gone through, and
Yeah.
very useful and valuable for students, valuable program, sir, it is.
Okay. Thank you, madam. So
Thank you.
madam, you can come at 4 p.m. So I have sent you 2 CV, so you have to brief
Yes,
them
sir,
about for vulnerability.
sure, sir, I will join back at 4 o'clock.
Okay. Thank you.
Welcome, sir. Thank you, sir.
Raju, sir. Now you are the host, sir.
Okay.
Yes, sir.
So we can leave the session as it is on.
Yes, sir. You can leave the session, sir.
No, I can leave it on, or I can
Yes,
come
sir.
out.
Yes, sir. Leave it on only, sir.
Okay. So all other participants of the oration competition will join around 1:15. So,
1:15,
okay,
sir.
thank you very
Then
much.
you can shut down, sir. No problem, sir. At 1:15,
No,
I
it's
will
okay. I will.
give the link. Same
I
link
will.
only, sir. Then I will
Okay.
make the host, sir.
Okay. No problem. Thank
Okay,
you.
sir. Okay, sir. Thank you, sir.
Excuse me, sir. Let us take a lunch break. In the post-lunch, we will have oral presentations divided into 4 different departments. They are pharmaceutics and regulatory affairs, pharmacology, cognosy, pharmaceutical analysis and chemistry, and pharmacy practice. So I request all the delegates, those who are received acceptance emails for your presentation, can join and present through the Google links shared in the WhatsApp groups and to your mails. Those who are willing to present now also, you can present their presentations in your interested thrust area and you can present. Those who are also interested to listen can also join to the presentations through the Google links. Thank you all. Let us meet in the afternoon session. Thank you so much.
What time it will start, madam?
Madam, it will start at 1:30 p.m. ma'am, as per the schedule.
1:30 as per the schedule. Thank you, madam.
Yes. Thank you. Thank you, ma'am. I request all the participants now leave the session and can join through the Google links for their
Google link. Okay.
presentations. Thank you.
question, but it's kind of very incomplete, or maybe I feel like I'm too much of it.